Welcome, friends. I'm so glad you're here. Whether you're drifting towards sleep, unwinding after a long day, or simply looking for a quiet companion, you found the right place. Before we begin, I'd love to know, where in the world are you listening from? Perhaps you're wrapped in a blanket, sitting by a softly lit window, or resting somewhere under an open sky. Wherever you are, I hope you're warm, comfortable, and ready to wander with me into Earth's most ancient and gentle fires. The cold light that burns without flame, without heat, without smoke. If you find peace or curiosity in these gentle stories, please consider liking the video, subscribing to the channel, or sharing it with someone who might need a moment of stillness too. And to those who've already joined along, thank you. 4 billion years ago, when Earth spins young and violent beneath an alien sky, something remarkable begins in the darkness of the first seas. The planet wears no familiar face yet, no continents as we know them, no breathable atmosphere, no life as we might recognize it. The oceans stretch endless and black under a methane-hazed atmosphere that filters sunlight into strange, orange twilight. But in the warm, mineral-rich waters that fill the lowest places of this restless world, chemistry discovers something extraordinary. It learns to make light. Not the harsh light of stars or the fleeting brilliance of lightning, but something softer. Something that glows without burning, that shines without consuming itself. In the primordial soup of Earth's earliest oceans, molecules begin their first tentative experiments with luminescence. They stumble upon combinations that transform chemical energy directly into photons, bypassing entirely the ancient contract that light must come paired with heat. Only took 4 billion years of random molecular collisions to figure out this particular trick. This discovery happens not once, but many times, in many places, in different corners of the warming seas. Various molecular arrangements independently find pathways to luminescence. Some involve copper-based compounds that pulse with blue-green radiance, their electrons cycling through excited states in precise quantum rhythms. Others rely on calcium-triggered reactions that flicker like underwater lightning, brief and brilliant and utterly cold. Still others employ iron-sulfur clusters that glow with deep red light, like embers that never warm. The chemistry is patient, methodical. It has, after all, geological time to perfect its craft. The early Earth provides an ideal laboratory for these experiments. Though perhaps laboratory suggests too much intention, the conditions are simply favorable, in the way that a warm, dark cave is favorable for sleeping. The atmosphere holds almost no oxygen, a gas that will later prove both essential and destructive to many forms of early luminescence. Without oxygen's eager interference, certain light-producing reactions can proceed with elegant simplicity. Hydrogen sulfide rises continuously from volcanic vents, providing the reducing conditions that some bioluminescent pathways require. Iron dissolved in seawater acts as a natural catalyst, enabling reactions that might otherwise need more complex molecular machinery. The temperature hovers in a narrow range that favors complex chemistry without destroying delicate molecular structures. Too hot. And the carefully folded proteins that enable luminescence unfold into useless tangles. Too cold, and the chemical reactions slow to geological paces, producing light so dim it barely qualifies as photons. But Earth's early oceans find the perfect middle ground, a planetary Goldilocks zone for the chemistry of cold fire. Perhaps most importantly, the early Earth is dark, profoundly, absolutely dark in ways that modern creatures can barely comprehend. The sun exists, of course, casting its radiation across the planet's surface during the day, but that surface bears little resemblance to the world we know. No land plants intercept and scatter sunlight into green filtered shadows. No ice caps reflect a brilliant white light back to space. No familiar blue sky creates the soft, diffuse illumination that seems so natural to us. Instead, The alien atmosphere absorbs and redirects solar radiation in wavelengths that paint everything in unfamiliar hues. But beneath even a few meters of water, permanent night reigns absolute. Any creature that develops in these depths, any cluster of self-replicating molecules that begins to resemble what we might generously call life, exists in a world without visual reference points. In such an environment, the ability to generate even the faintest glow represents a tremendous evolutionary advantage. It is the difference between navigating by touch and navigating by sight, between random encounters and purposeful movement. The first organisms to master this cold fire are almost incomprehensibly simple by modern standards. They are barely more than organized chemical reactions wrapped in lipid membranes. Molecular conspiracies that have learned to persist and replicate themselves in the face of thermodynamic entropy. But they possess something their neighbors lack: the molecular machinery to convert nutrients directly into visible light. This machinery, which scientists will eventually name luciferase after the Roman bearer of light, represents one of evolution's most elegant solutions to an ancient problem. How does one signal, hunt, or navigate in absolute darkness without wasting precious energy on heat? The answer lies in the quantum mechanical properties of certain organic molecules. Though to describe these early organisms as understanding quantum mechanics would be like crediting a falling apple with understanding gravity. When electrons in these specialized molecules absorb energy from ATP, from specific chemical substrates, from the breakdown of simple nutrients, they jump to higher energy states with the predictable enthusiasm of excited atoms everywhere. But unlike most molecular processes, which release this energy as heat or mechanical motion, these particular molecules have stumbled upon a way to release it as pure light. The electrons cascade back to their ground state in precise quantum steps, each step corresponding to a specific wavelength of visible radiation. Blue light emerges when the energy gaps are large, red when they are smaller, green when they fall somewhere in between. The process is remarkably efficient. Some biological light sources convert nearly 90% of their input energy directly into photons, an efficiency that makes modern LED bulbs look positively wasteful. What emerges in Earth's ancient seas is not random flickering, but organized, purposeful luminescence that suggests even the simplest life possesses something approaching intentionality. Some early organisms glow continuously, like living nightlights in the oceanic dark, their steady radiance marking their presence to anyone equipped to see. Others pulse rhythmically, their light production tied to internal biological clocks that tick in harmony with chemical cycles we are only beginning to understand. The rhythm might follow the breakdown of nutrients, the accumulation of waste products, or cycles of cellular division that repeat with the reliability of geological processes. Still others remain dark until disturbed, then flare briefly in startled brilliance before fading back to invisibility. These represent perhaps the most sophisticated early response to predation, the biological equivalent of a burglar alarm that floods the scene with light, startling attackers and potentially attracting their predators in turn. Only took a few 100 million years to invent the concept of calling for help. The biochemistry underlying these displays varies remarkably even among the earliest practitioners, suggesting that the problem of making cold light admits many different solutions. Some rely on luciferin molecules, small, energy-rich compounds that burn cold when combined with oxygen and the right enzymes. The luciferin-luciferase reaction will prove so successful that nature will return to it again and again, refining it across geological ages until it appears in organisms as diverse as fireflies and deep-sea fish. Others use different chemical pathways entirely, generating light through reactions that involve no oxygen at all. These anaerobic light sources rely instead on different electron acceptors, nitrates, sulfates, even ion compounds that cycle through various oxidation states. The diversity, suggests that bioluminescence did not evolve once and spread like a biological innovation, but rather emerged independently multiple times as life explored different solutions to the same fundamental challenge: how to make light without fire. The energy economics of this early luminescence are surprisingly favorable, though the accounting must be done in currencies we rarely consider. While producing light does require metabolic investment, ATP must be spent, substrates must be consumed, specialized enzymes must be manufactured and maintained. The payoff often justifies the cost in ways that compound over generations. A glowing organism can attract food by mimicking the chemical signatures that smaller organisms find irresistible. It can confuse predators by creating visual illusions in the dark water. It can locate mates across vast stretches of empty ocean, turning random encounters into purposeful reproductive events. More subtly, luminescence can serve as a form of cellular communication, allowing simple organisms to coordinate their behavior across populations numbering in the billions. When one cell encounters food or danger, its light can signal others to approach or flee. Entire populations can synchronize their activities through networks of luminescent communication that prefigure, by billions of years, the electronic networks that modern civilizations depend upon. Yet mysteries abound in these ancient seas, questions that remain tantalizingly unresolved despite decades of careful research. How do the first light-producing organisms prevent their own luminescence from attracting unwanted attention? What determines the specific colors of their glow? The deep blues that penetrate furthest through water, the jade greens that stand out against most backgrounds, the occasional flashes of yellow and red that seem almost decorative in their rarity. Why do some species pulse while others shine steadily? And what internal mechanisms control these different temporal patterns? The answers remain partially hidden in the geological record, obscured by the simple fact that soft biochemical machinery rarely fossilizes in recognizable form, unlike bones or shells, which preserve their mineral structures across geological time. The delicate proteins and enzymes that enable luminescence decompose and vanish. Scientists must instead piece together this ancient story from the molecular evidence preserved in modern organisms, reading backwards through genetic sequences to reconstruct what life's earliest experiments with light might have looked like. It is detective work of the most challenging kind, inferring ancient history from modern biology, tracing evolutionary pathways that branch and merge across billions of years. But the evidence, fragmentary as it is, points toward a remarkable conclusion. The basic chemical principles discovered in those first glowing cells remain essentially unchanged in organisms living today. A modern firefly's flash and a bacterial cell's steady glow both rely on molecular machinery that traces its ancestry back to these primordial seas. In the darkness of 4 billion years ago, life makes its first tentative announcement of presence, not through sound, which requires medium and listener, not through movement, which might go unnoticed in the vastness, not through chemical signals that diffuse and fade, but through light itself, cold, efficient, purposeful light that cuts through the surrounding blackness without generating a single degree of unnecessary heat. The candle that burns without fire has been lit, and it will never be extinguished. Around these first glowing cells, the ancient ocean continues its slow chemical evolution, indifferent to the revolutionary events taking place in its depths. Minerals precipitate and dissolve according to temperature and pH. Volcanic gases bubble up from the sea floor, carrying with them the chemical signatures of the planet's molten interior. Tides rise and fall under the influence of a moon that hangs much closer to Earth than it does today, creating more dramatic tidal cycles that stir the primitive seas into constant, gentle motion. But in the depths, where light has never before existed except as the briefest flash of volcanic lightning, Something fundamentally new has entered the world. Life has learned to shine. And in learning to shine, it has taken its first step toward the complex biochemical symphonies that will eventually fill every corner of the planet with cold fire. The bacterial lanterns emerge from the chemical darkness like scattered stars finding their voices for the first time. These are not the complex, multicellular organisms that will later master bioluminescence with such spectacular artistry, but something far simpler and, in its own way, more remarkable. Single cells, barely visible to any eye that might exist to see them, learn to transform the basic chemistry of survival into steady, purposeful light. The bacteria of the Archean seas exist at scales that make their achievements all the more extraordinary. Each cell measures perhaps 1 micrometer across, so small that millions could fit comfortably on the head of a pin. If pins existed, if heads existed, if the concept of comfortable fitting had any meaning in their microscopic realm, yet within these infinitesimal volumes, they construct and operate luminescent machinery that rivals anything evolution will later produce. They are living proof that sophistication need not correlate with size, that the most profound innovations often emerge from the most constrained circumstances. The biochemistry they employ follows pathways that seem almost impossibly direct in their efficiency. A typical luminescent bacterium synthesizes its luciferin substrates through metabolic routes that branch off from the same chemical networks used for basic cellular maintenance. The cell takes in simple organic compounds, perhaps acetate molecules scavenged from the breakdown of other organisms, perhaps amino acids absorbed from the surrounding water, and through a series of carefully orchestrated enzymatic reactions, converts them into the light-producing compounds that will define its relationship with the larger world. The luciferase enzymes themselves represent masterpieces of molecular architecture, proteins that fold into precise three-dimensional configurations optimized for a single, specific purpose, catalyzing the controlled oxidation of luciferin in ways that maximize light output while minimizing energy waste. The active sites of these enzymes position reactant molecules with atomic precision, holding luciferin and oxygen in exactly the right orientations to ensure that their chemical reaction proceeds along pathways that favor photon emission over heat production. Each bacterial cell can contain thousands of luciferase molecules, distributed throughout its cytoplasm like molecular streetlights, illuminating the biochemical pathways of life. When conditions are favorable, when substrates are abundant, when energy reserves are adequate, when the cellular machinery operates within optimal parameters, these enzymes work in coordinated symphony, producing steady streams of photons that emerge from the cell as visible light. The colors these bacterial lanterns produce depend on subtle details of molecular structure that reflect millions of years of chemical refinement. Most early bacteria emit blue-green light, wavelengths that penetrate seawater with minimal absorption and scattering. The specific shade, whether more blue or more green, whether pure or tinged with other colors, depends on how luciferase molecules position luciferin during the critical moments of oxidation. Small changes in amino acid sequence can alter the enzyme's active site geometry, shifting emission spectra in ways that might seem trivial but prove crucial for survival in environments where even slight advantages compound over geological time. Some bacterial species develop the ability to modulate their light output in response to population density, a phenomenon that prefigures by billions of years the complex social behaviors of higher organisms. As cell numbers increase in a given volume of water, the concentration of certain signaling molecules rises proportionally. When these molecules reach threshold levels, they trigger genetic circuits that increase luciferase production, causing the entire bacterial population to glow more brightly. The result is a form of primitive communication that allows single-celled organisms to coordinate their activities across vast spatial scales. This quorum sensing, as scientists will eventually term it, enables bacterial populations to achieve collective behaviors that would be impossible for individual cells. When enough bacteria accumulate in a particular location, their coordinated luminescence can create patches of glowing water visible from considerable distances. These bacterial beacons may serve to attract other microorganisms that the bacteria prey upon, or to repel predators that avoid well-illuminated areas, or simply to maintain population cohesion in the vast, dark expanses of the primitive oceans. The energy economics of bacterial luminescence involve calculations of remarkable subtlety for organisms that possess no nervous systems, no capacity for conscious deliberation, no ability to weigh costs against benefits in any sense we might recognize. Yet the persistence of luminescent bacteria across geological time suggests that natural selection has repeatedly found the energy investment worthwhile. Each photon produced requires the expenditure of cellular resources that might otherwise be used for growth or reproduction. The fact that luminescent bacteria continue to invest in light production implies that the benefits, whatever they might be, consistently outweigh the metabolic costs. These benefits likely vary depending on environmental conditions and ecological context. In nutrient-rich environments, where energy is abundant and competition is intense, luminescence might serve primarily as a form of advertisement, helping bacteria attract symbiotic partners or organic matter that serves as both food and raw material for further light production. In nutrient-poor waters, where every molecule of ATP represents precious metabolic currency, luminescence might function more like a biological insurance policy, a way of maintaining visibility and mobility in environments where most organisms remain dormant. The temporal patterns of bacterial luminescence reveal additional layers of complexity in organisms that otherwise appear remarkably simple. Some species maintain steady light output as long as conditions permit, their glow as constant as the biochemical reactions that sustain it. Others pulse rhythmically, their luminescence tied to internal clocks that track cellular division cycles, metabolic rhythms, or the availability of key substrates. Still others respond to external stimuli with changes in light intensity that suggest primitive forms of environmental sensing and response. The mechanisms underlying these temporal variations involve regulatory networks that control when and how much luciferase the bacteria produce. Gene expression responds to chemical signals that reflect the cell's internal state and external environment. When nutrients are abundant, genes coding for luminescent proteins are transcribed at high rates, flooding the cell with the molecular machinery needed for light production. When resources become scarce, these same genes are downregulated, allowing the cell to conserve energy for more essential functions while maintaining just enough luminescent capacity to preserve the option of returning to full brightness when conditions improve. Yet mysteries persist in bacterial luminescence that continue to intrigue researchers billions of years after these first microbial lanterns began to glow. Why do some bacterial species produce light continuously while others restrict luminescence to specific phases of their growth cycles? What determines the precise wavelengths individual species emit? And how do these spectral characteristics relate to the ecological niches they occupy? How do bacteria coordinate their luminescence across populations that span kilometers of ocean water? And what information do they encode in the temporal patterns of their collective glow? The answers remain partially hidden in the molecular details of bacterial physiology, encrypted in genetic sequences that have been modified and refined across countless generations. Each luminescent bacterium carries within its chromosome the accumulated wisdom of billions of years of chemical experimentation, the molecular memory of countless successful adaptations to the challenging conditions of the primitive Earth. Their light represents not just a biological function, but a form of evolutionary testimony, evidence of the remarkable solutions that life discovers when faced with the fundamental problem of visibility in an utterly dark world. In the depths of the Archean oceans, these bacterial lanterns continue their patient work, converting simple organic molecules into streams of cold photons with efficiency that would impress modern engineers. They glow not because they choose to glow, not because they understand the benefits of luminescence, but because the chemistry of their existence has been shaped by selective pressures that favor organisms capable of making light. Their illumination represents pure function distilled into its most essential form. Biology reduced to the fundamental task of transforming chemical energy into visible signals that can be detected, interpreted, and acted upon by other living systems. Each bacterial cell that glows in the darkness of the early Earth contributes its small portion of photons to the growing constellation of biological light that will eventually transform the planet's relationship with illumination. They are the pioneers of a technology that will spread through every major branch of the tree of life, the inventors of biochemical principles that will find expression in organisms as diverse as mushrooms and fireflies and deep-sea fish. Only took 4 billion years for bacteria to light the first candle that burns without fire, and in lighting it, they illuminate pathways toward forms of life that seem almost impossibly complex by comparison, yet depend entirely on the same fundamental chemistry these simple cells have already mastered. The bacterial lanterns emerge from the chemical darkness like scattered stars finding their voices for the first time. These are not the complex, multicellular organisms that will later master bioluminescence with such spectacular artistry, but something far simpler and, in its own way, more remarkable: single cells, barely visible to any eye that might exist to see them, learn to transform the basic chemistry of survival into steady, purposeful light. The bacteria of the Archean seas exist at scales that make their achievements all the more extraordinary. Each cell measures perhaps 1 micrometer across, so small that millions could fit comfortably on the head of a pin. If pins existed, if heads existed, if the concept of comfortable fitting had any meaning in their microscopic realm, yet within these infinitesimal volumes, they construct and operate luminescent machinery that rivals anything evolution will later produce. They are living proof that sophistication need not correlate with size, that the most profound innovations often emerge from the most constrained circumstances. The biochemistry they employ follows pathways that seem almost impossibly direct in their efficiency. A typical luminescent bacterium synthesizes its luciferin substrates through metabolic routes that branch off from the same chemical networks used for basic cellular maintenance. The cell takes in simple organic compounds, perhaps acetate molecules scavenged from the breakdown of other organisms, perhaps amino acids absorbed from the surrounding water, and through a series of carefully orchestrated enzymatic reactions, converts them into the light-producing compounds that will define its relationship with the larger world. The luciferase enzymes themselves represent masterpieces of molecular architecture, proteins that fold into precise three-dimensional configurations optimized for a single, specific purpose, catalyzing the controlled oxidation of luciferin in ways that maximize light output while minimizing energy waste. The active sites of these enzymes position reactant molecules with atomic precision, holding luciferin and oxygen in exactly the right orientations to ensure that their chemical reaction proceeds along pathways that favor photon emission over heat production. Each bacterial cell can contain thousands of luciferase molecules, distributed throughout its cytoplasm like molecular streetlights, illuminating the biochemical pathways of life. When conditions are favorable, when substrates are abundant, when energy reserves are adequate, when the cellular machinery operates within optimal parameters, these enzymes work in coordinated symphony, producing steady streams of photons that emerge from the cell as visible light. The colors these bacterial lanterns produce depend on subtle details of molecular structure that reflect millions of years of chemical refinement. Most early bacteria emit blue-green light, wavelengths that penetrate seawater with minimal absorption and scattering. The specific shade, whether more blue or more green, whether pure or tinged with other colors, depends on how luciferase molecules position luciferin during the critical moments of oxidation. Small changes in amino acid sequence can alter the enzyme's active site geometry, shifting emission spectra in ways that might seem trivial but prove crucial for survival in environments where even slight advantages compound over geological time. Some bacterial species develop the ability to modulate their light output in response to population density, a phenomenon that prefigures by billions of years the complex social behaviors of higher organisms. As cell numbers increase in a given volume of water, the concentration of certain signaling molecules rises proportionally. When these molecules reach threshold levels, they trigger genetic circuits that increase luciferase production, causing the entire bacterial population to glow more brightly. The result is a form of primitive communication that allows single-celled organisms to coordinate their activities across vast spatial scales. This quorum sensing, as scientists will eventually term it, enables bacterial populations to achieve collective behaviors that would be impossible for individual cells. When enough bacteria accumulate in a particular location, their coordinated luminescence can create patches of glowing water visible from considerable distances. These bacterial beacons may serve to attract other microorganisms that the bacteria prey upon, or to repel predators that avoid well-illuminated areas, or simply to maintain population cohesion in the vast, dark expanses of the primitive oceans. The energy economics of bacterial luminescence involve calculations of remarkable subtlety for organisms that possess no nervous systems, no capacity for conscious deliberation, no ability to weigh costs against benefits in any sense we might recognize. Yet the persistence of luminescent bacteria across geological time suggests that natural selection has repeatedly found the energy investment worthwhile. Each photon produced requires the expenditure of cellular resources that might otherwise be used for growth or reproduction. The fact that luminescent bacteria continue to invest in light production implies that the benefits, whatever they might be, consistently outweigh the metabolic costs. These benefits likely vary depending on environmental conditions and ecological context. In nutrient-rich environments, where energy is abundant and competition is intense, luminescence might serve primarily as a form of advertisement, helping bacteria attract symbiotic partners or organic matter that serves as both food and raw material for further light production. In nutrient-poor waters, where every molecule of ATP represents precious metabolic currency, luminescence might function more like a biological insurance policy, a way of maintaining visibility and mobility in environments where most organisms remain dormant. The temporal patterns of bacterial luminescence reveal additional layers of complexity in organisms that otherwise appear remarkably simple. Some species maintain steady light output as long as conditions permit, their glow as constant as the biochemical reactions that sustain it. Others pulse rhythmically, their luminescence tied to internal clocks that track cellular division cycles, metabolic rhythms, or the availability of key substrates. Still, others respond to external stimuli with changes in light intensity that suggest primitive forms of environmental sensing and response. The mechanisms underlying these temporal variations involve regulatory networks that control when and how much luciferase the bacteria produce. Gene expression responds to chemical signals that reflect the cell's internal state and external environment. When nutrients are abundant, genes coding for luminescent proteins are transcribed at high rates, flooding the cell with the molecular machinery needed for light production. When resources become scarce, these same genes are downregulated, allowing the cell to conserve energy for more essential functions while maintaining just enough luminescent capacity to preserve the option of returning to full brightness when conditions improve. Yet mysteries persist in bacterial luminescence that continue to intrigue researchers billions of years after these first microbial lanterns began to glow. Why do some bacterial species produce light continuously while others restrict luminescence to specific phases of their growth cycles? What determines the precise wavelengths individual species emit? And how do these spectral characteristics relate to the ecological niches they occupy? How do bacteria coordinate their luminescence across populations that span kilometers of ocean water? And what information do they encode in the temporal patterns of their collective glow? The answers remain partially hidden in the molecular details of bacterial physiology, encrypted in genetic sequences that have been modified and refined across countless generations. Each luminescent bacterium carries within its chromosome the accumulated wisdom of billions of years of chemical experimentation, the molecular memory of countless successful adaptations to the challenging conditions of the primitive Earth. Their light represents not just a biological function, but a form of evolutionary testimony, evidence of the remarkable solutions that life discovers when faced with the fundamental problem of visibility in an utterly dark world. In the depths of the Archean oceans, these bacterial lanterns continue their patient work, converting simple organic molecules into streams of cold photons with efficiency that would impress modern engineers. They glow not because they choose to glow, not because they understand the benefits of luminescence, but because the chemistry of their existence has been shaped by selective pressures that favor organisms capable of making light. Their illumination represents pure function distilled into its most essential form, biology reduced to the fundamental task of transforming chemical energy into visible signals that can be detected, interpreted, and acted upon by other living systems. Each bacterial cell that glows in the darkness of the early Earth contributes its small portion of photons to the growing constellation of biological light that will eventually transform the planet's relationship with illumination. They are the pioneers of a technology that will spread through every major branch of the tree of life, the inventors of biochemical principles that will find expression in organisms as diverse as mushrooms and fireflies and deep-sea fish. Only took 4 billion years for bacteria to light the first candle that burns without fire, and in lighting it, they illuminate pathways toward forms of life that seem almost impossibly complex by comparison, yet depend entirely on the same fundamental chemistry these simple cells have already mastered. The ocean becomes a liquid constellation as dinoflagellates spread across the prehistoric seas, their microscopic bodies carrying the ancient chemistry of cold fire into new realms of possibility. These are not bacteria, though they share the seas with those first bacterial lanterns. Instead, they represent something more complex, something that points toward the elaborate architectures of cellular organization that will eventually give rise to all visible life. Yet they remain, by any modern standard, profoundly simple. Single cells that drift with the currents, harvesting sunlight and nutrients with equal efficiency while maintaining their capacity to transform chemical energy into streams of visible photons. The dinoflagellates emerge during the long, slow transition when Earth's chemistry begins its gradual shift towards something more familiar. Oxygen starts to accumulate in the atmosphere, though still in concentrations that would be lethal to most modern organisms. The iron dissolved in seawater begins to precipitate out in great bands of rust-red sediment that will eventually become the banded iron formations geologists use to read the chemical history of these ancient times. The very composition of seawater itself evolves as volcanic outgassing and weathering processes establish new equilibria between the atmosphere, the hydrosphere, and the solid earth beneath. In this changing world, dinoflagellates develop luminescent capabilities that surpass those of their bacterial predecessors in both sophistication and spectral beauty. Their light-producing organelles, called syntions, represent marvels of subcellular architecture, membrane-bound compartments that contain all the molecular machinery needed for luminescence in precisely organized, optimally efficient configurations. Each scintillon houses its own complement of luciferin substrates, luciferase enzymes, and the cofactors necessary to sustain light production. More remarkably, these organelles can be triggered to discharge their luminescent contents in coordinated bursts that flood the surrounding water with blue-green radiance. The triggering mechanism itself reveals unexpected complexity in organisms that possess no nervous systems, no centralized control structures, no apparent capacity for decision-making in any sense we might recognize. When a dinoflagellate experiences mechanical disturbance, a change in water pressure, a collision with another organism, a sudden shift in chemical concentration, the stimulation propagates through the cell's membrane systems as a wave of electrical depolarization. This electrical signal, carried by the movement of ions across lipid bilayers, reaches the syntions and causes them to undergo rapid structural changes that release their stored light-producing compounds into the surrounding cytoplasm. The chemistry that follows unfolds with the precision of a molecular ballet choreographed by billions of years of evolutionary refinement. Luciferin molecules encounter luciferase enzymes in the presence of oxygen that has diffused into the cell from the surrounding seawater. The familiar oxidative reaction proceeds, but now it takes place in cellular environments that can regulate pH ion concentrations and the availability of cofactors with far greater precision than any bacterial system. The result is light production that can be controlled, modulated, and optimized in ways that open entirely new possibilities for biological function. Individual dinoflagellate cells can produce light intensities that seem almost impossible given their microscopic size. A single cell, barely visible to the naked eye, can generate flashes bright enough to be detected by organisms many times larger, swimming many body lengths away. The secret lies not just in the efficiency of the light-producing chemistry, but in the temporal concentration of the luminescent reaction. Rather than glowing steadily like many bacteria, dinoflagellates compress their light output into brief, intense pulses that maximize visibility while minimizing the total energy investment required. These flashes serve multiple functions that demonstrate the remarkable versatility of bioluminescence as an evolutionary strategy. When disturbed by potential predators, a dinoflagellate's sudden burst of light may startle the predator long enough for the cell to escape. The illumination might also attract secondary predators that pose threats to the original predator, turning the light-producing dinoflagellate into bait that draws its enemy's enemies into defensive range. Still, other functions remain mysterious. Some dinoflagellates flash in response to stimuli that seem unrelated to predation, suggesting that their luminescence serves purposes we do not yet fully understand. The collective behavior of dinoflagellate populations creates some of the most spectacular displays of bioluminescence in Earth's history. When millions or billions of these microscopic organisms concentrate in particular regions of the ocean, their coordinated flashing can transform entire stretches of seawater into liquid fireworks. Each individual cell responds to disturbance by producing its brief flash. But when vast populations are disturbed simultaneously by wind-driven waves, by the passage of larger organisms, by sudden changes in water chemistry, the result is a coordinated light show that can be visible from great distances. These mass displays serve functions that extend far beyond the survival needs of individual cells. The coordinated luminescence of dinoflagellate blooms may help maintain population cohesion by providing visual cues that allow dispersed individuals to locate and rejoin the main group. The light may also serve as a form of chemical communication, since many dinoflagellates produce bioluminescence in response to the same chemical signals they use for other forms of intercellular coordination. When one cell detects the presence of certain organic compounds that indicate food, danger, or reproductive opportunity, Its luminescent response may signal this information to neighboring cells across distances far greater than chemical diffusion alone could bridge. The wavelengths dinoflagellates emit reflect both the constraints of their biochemistry and the optical properties of their marine environment. Most produce light in the blue-green portion of the spectrum, wavelengths that travel furthest through seawater with minimal absorption or scattering. This spectral choice represents an elegant solution to the physics of underwater communication by producing light that the ocean itself transmits most efficiently. Dinoflagellates maximize the distance their signals can travel while minimizing the energy required to generate them. Yet variations exist that hint at the extraordinary diversity of strategies these organisms have evolved. Some dinoflagellate species produce light that shifts subtly toward the blue end of the spectrum, wavelengths that may be less visible to certain types of predators while remaining detectable to potential prey or symbiotic partners. Others emit light that includes small amounts of longer wavelengths, creating subtle color variations that might encode different types of information for organisms capable of detecting such spectral nuances. The regulation of luminescence in dinoflagellates involves molecular mechanisms that prefigure the complex regulatory networks found in more advanced organisms. Gene expression responds to environmental cues in sophisticated ways that allow cells to adjust their light-producing capacity based on factors like nutrient availability, population density, and the time of day. Some species show circadian rhythms in their luminescent capacity, producing more light during certain hours in patterns that suggest coordination with the daily cycles of predation and feeding that structure marine ecosystems. The evolutionary success of luminescent dinoflagellates demonstrates the remarkable power of bioluminescence as an adaptive strategy. These organisms have spread to virtually every marine environment where conditions permit their survival, from tropical surface waters to cold polar seas, from nutrient-rich coastal regions to the sparse biological deserts of the open ocean. Their luminescence allows them to thrive in ecological niches that might otherwise be closed to organisms of their size and metabolic capabilities. Yet mysteries remain embedded in the quantum mechanical processes that enable dinoflagellate luminescence. Why do some species flash once when disturbed, while others produce trains of repeated pulses? What determines the precise timing of luminescent responses? And how do cells coordinate their flashing across populations that span vast distances? How do the molecular details of luciferase chemistry vary among different dinoflagellate lineages? And what do these variations reveal about the evolutionary history of bioluminescence itself? In the depths of the prehistoric seas, dinoflagellates continue their patient work of converting mechanical and chemical stimuli into streams of cold photons that cut through the surrounding darkness like biological lightning. Their flashes illuminate not just the water around them, but the pathways of evolutionary possibility that will eventually lead to all the spectacular forms of bioluminescence that grace the modern world. Only took 1 billion years for these microscopic organisms to perfect the art of liquid fireworks. And in perfecting it, they establish principles of biological light production that will persist, unchanged in their fundamental chemistry. Through all the subsequent elaborations of life on Earth, the jellies learn to burn with a patience that spans geological epochs, their translucent bodies drifting through the Precambrian depths like living lanterns crafted from organized water. These ancient cnidarians represent something fundamentally different from the bacterial and dinoflagellate lights that preceded them, the first multicellular organisms to master the chemistry of cold fire, the pioneers of bioluminescence as a coordinated tissue-level phenomenon rather than the solitary glow of individual cells. Their bodies consist of little more than organized water held in place by delicate protein scaffolds. Yet within this seemingly simple architecture, they construct luminescent systems of remarkable sophistication. Specialized photocytes, cells dedicated entirely to light production, cluster along the margins of their bell-shaped bodies. They're luciferin and luciferase concentrated in organelles that can discharge coordinated flashes visible across vast distances in the dark water. The jelly's luminescence emerges not from the random firing of individual cells, but from carefully orchestrated displays that suggest primitive forms of neural coordination millions of years before anything resembling a brain evolves. The biochemistry these ancient cnidarians employ builds upon the same fundamental principles discovered by bacteria and dinoflagellates, yet elaborates them in ways that reveal new possibilities for biological light production. Their luciferin, which scientists will eventually identify as coelenterazine, represents a molecular architecture optimized for the specific challenges of multicellular luminescence. Unlike the simpler compounds used by bacteria, coelenterazine can be stored in specialized vesicles for extended periods, then released in precisely controlled quantities when luminescence is required. This biochemical innovation allows jellies to produce light on demand rather than maintaining the steady glow that characterizes many bacterial systems. The luciferase enzymes that catalyze coelenterazine oxidation show remarkable diversity across different cnidarian lineages, reflecting millions of years of independent evolution and optimization. Some variants produce blue light that penetrates deepest into seawater, while others emit green wavelengths that may be more visible to certain types of prey or predators. Still others generate complex spectra that include multiple wavelengths, creating light displays of subtle beauty that seem almost decorative until one considers their probable functions in the harsh economy of survival and reproduction. The spatial organization of luminescence within jellyfish tissues reveals unexpected sophistication in organisms that lack most of the organ systems we associate with complex life. Photocytes arrange themselves in precise patterns along the bell margin, their distribution optimized to create specific types of light displays. Some jellies concentrate their luminescent cells in discrete spots that flash independently, creating moving constellations of light as different regions of the organism respond to various stimuli. Others distribute their photocytes more evenly, producing diffuse illumination that transforms the entire animal into a living lamp suspended in the darkness. The neural networks that control these displays represent some of the earliest experiments in coordinated multicellular behavior. Primitive nerve nets spread through jellyfish tissues like biological electrical circuits, carrying signals that synchronize the firing of photocytes across the entire organism. When one part of a jellyfish detects a threat or opportunity, the stimulus propagates through this neural network as waves of electrical activity that trigger coordinated luminescent responses. The result is light production that can be modulated, directed, and timed with precision that far exceeds what individual cells could achieve working independently. These luminescent displays serve functions that demonstrate the remarkable versatility of bioluminescence as multicellular organisms explore new ecological strategies. The most obvious function involves predator deterrence when threatened. Many jellies produce startling flashes of light that may confuse or frighten potential predators long enough for escape. But the light also serves more subtle purposes that hint at the complex behavioral repertoires these seemingly simple organisms possess. Some jellies use luminescence to attract prey, creating light displays that mimic the bioluminescent signatures of organisms their prey typically consume. The evolutionary success of luminescent cnidarians transforms the optical environment of the ancient oceans in profound ways. For the first time in Earth's history, significant quantities of biological light emanate from multicellular organisms large enough to be visible to other creatures across considerable distances. The seas begin to develop optical landscapes, regions where bioluminescent organisms create patterns of illumination that can be navigated, interpreted, and exploited by other forms of life. Only took 500 million years for biology to invent natural lighting systems. The energy economics of multicellular luminescence involve considerations that extend far beyond the simple cost-benefit calculations that govern bacterial bioluminescence. Jellies must invest cellular resources not just in the production of luciferin and luciferase, but in the construction and maintenance of specialized photosites, the neural networks that control them, and the sophisticated vesicle systems that store and release light-producing compounds. The total metabolic cost of maintaining luminescent capability in a multicellular organism exceeds that required by single cells by orders of magnitude. Yet the benefits scale proportionally, creating new categories of biological advantage that are simply unavailable to unicellular organisms. A luminescent jellyfish can create light displays visible across distances measured in hundreds of meters, rather than the micrometers or millimeters that limit bacterial and dinoflagellate communication. They can modulate their luminescence in complex temporal patterns that encode different types of information for different audiences. They can coordinate their light production with other behaviors, feeding, reproduction, migration, in ways that maximize the effectiveness of each activity. The temporal dynamics of cnidarian luminescence reveal additional layers of complexity that distinguish multicellular light production from its simpler predecessors. Some jellies flash briefly when disturbed, producing single pulses of light that serve as burglar alarms to startle predators or attract their enemies. Others generate trains of repeated flashes, creating rhythmic displays that may serve communicative functions we do not yet fully understand. Still others can maintain steady illumination for extended periods, transforming themselves into biological beacons that remain visible across vast expanses of dark water. The control mechanisms underlying these different patterns involve neural circuits that represent early experiments in biological computation. Simple networks of interconnected neurons process sensory information and generate motor outputs that coordinate luminescent responses with other behaviors. When a jellyfish encounters prey, its nervous system must integrate information about the prey's size, speed, and direction of movement, then coordinate appropriate responses that might include changes in swimming behavior, deployment of stinging cells, and specific patterns of luminescent display. The biochemical diversity of cnidarian luminescence systems reflects the remarkable evolutionary creativity with which these organisms have explored the possibilities of biological light production. Different species employ variations of the basic coenterozyme luciferase reaction that produce different colors, intensities, and temporal patterns of illumination. Some have evolved multiple types of photosites within the same organism, each specialized for different types of display. Others have developed mechanisms for recycling luminescent compounds, creating closed loops of light production that minimize the metabolic costs of maintaining bioluminescent capability. Yet mysteries persist in cnidarian luminescence that continue to intrigue scientists hundreds of millions of years after these first multicellular lanterns began to glow. How do the neural networks that control luminescence integrate with other behavioral systems to create coordinated responses to environmental challenges? What determines the specific patterns of photosite distribution within different species? And how do these patterns relate to the ecological functions of their light displays? How do cnidarians regulate the production and storage of luciferin compounds to maintain optimal luminescent capacity under varying environmental conditions? The answers remain encrypted in the molecular details of cnidarian physiology and the evolutionary history encoded in their genetic sequences. Each luminescent jellyfish carries within its genome the accumulated wisdom of countless generations of natural selection, the molecular memory of successful solutions to the fundamental challenges of survival in a dark and dangerous world. Their light represents not just a biological function, but a form of evolutionary testimony to the remarkable innovations that emerge when multicellular life begins to explore the possibilities of coordinated behavior. In the depths of the Precambrian seas, Ancient Cyanidarians continue their patient experimentation with the chemistry of cold fire, their translucent bodies pulsing through dark waters like living galaxies sustained by the same fundamental biochemistry that enables all biological luminescence. They glow not because they understand the physics of photon emission or the evolutionary advantages of visibility, but because the molecular machinery of their cells has been shaped by selective pressures that favor organisms capable of transforming chemical energy into precisely controlled displays of visible light. Their luminescence illuminates pathways toward forms of biological complexity that seem almost unimaginably elaborate by comparison, yet depend entirely on the same quantum mechanical processes these simple organisms have already mastered with such elegant efficiency. Symbiosis of light emerges as bacteria, and larger organisms discover the profound advantages of sharing both the burden and the beauty of bioluminescence. In the ancient seas where individual survival demands constant innovation, some organisms begin to experiment with arrangements that seem almost impossibly cooperative. Bacteria that live within the tissues of larger creatures, providing steady illumination in exchange for nutrients and protection, while their hosts gain access to biological lighting systems without investing in the complex biochemical machinery required to produce light independently. These partnerships represent one of evolution's most elegant solutions to the metabolic costs of luminescence. A fish or squid that attempts to synthesize its own luciferin and luciferase must dedicate significant genetic and cellular resources to manufacturing, storing, and regulating light-producing compounds. But an organism that provides suitable habitat for luminescent bacteria can outsource this entire biochemical enterprise to specialized microbial partners that have already perfected the relevant chemistry through millions of years of independent evolution. The bacterial partners in these relationships are descendants of the first bacterial lanterns, organisms whose light-producing capabilities have been refined through countless generations of selection for efficiency and reliability. They bring to these partnerships not just the ability to produce light, but sophisticated regulatory mechanisms that allow luminescence to be controlled, modulated, and coordinated with the needs of their multicellular hosts. The bacteria can adjust their light output in response to chemical signals from host tissues, creating integrated lighting systems that function as seamlessly as if they were products of a single genome. The anatomical arrangements that accommodate these bacterial symbionts reveal remarkable innovations in tissue organization and physiological integration. Some hosts develop specialized light organs, complex structures that house bacterial colonies in carefully controlled environments optimized for both microbial survival and light production. These organs often include sophisticated optical systems with lenses, reflectors, and shutters that can focus, direct, and modulate the bacterial illumination with precision that rivals engineered lighting devices. The chemistry of these symbiotic relationships involves intricate negotiations between bacterial metabolism and host physiology that must be sustained across the lifetimes of both partners. Hosts must provide their bacterial symbionts with steady supplies of nutrients, typically simple organic compounds that the bacteria can metabolize to fuel their luminescent reactions. In return, the bacteria must regulate their light production to serve their host's needs rather than simply maximizing their own survival and reproduction. This regulation involves molecular communication systems that allow hosts and symbionts to coordinate their activities through chemical signals that cross species boundaries. When a host requires increased illumination, perhaps to attract prey, communicate with potential mates, or confuse predators, it releases specific compounds that bacterial symbionts recognize and respond to by increasing their light output. When illumination is not needed, different chemical signals can reduce or eliminate bacterial luminescence, conserving energy for both partners. The evolutionary origins of these partnerships remain partially mysterious. embedded in ancient history that left few fossil traces of the delicate negotiations between hosts and symbionts. What seems likely is that the relationships began as more casual associations, bacteria that happened to colonize the tissues of larger organisms, perhaps initially as parasites or simply as opportunistic inhabitants of favorable microenvironments. Over time, those associations that proved mutually beneficial would have been favored by natural selection, gradually evolving into the sophisticated partnerships that characterize modern symbiotic luminescence. The success of these arrangements has led to their independent evolution across numerous lineages of marine organisms. Fish, squid, crustaceans, and various other creatures have all discovered the advantages of bacterial partnerships, often developing remarkably similar solutions to the challenges of housing and managing microbial symbionts. This convergent evolution suggests that symbiotic luminescence represents a fundamentally advantageous strategy, sufficiently beneficial that natural selection has repeatedly favored its development despite the considerable challenges involved in establishing and maintaining interspecies partnerships. The bacterial partners themselves often undergo significant evolutionary changes as they adapt to life within their hosts. Free-living luminescent bacteria must maintain the full complement of genes and metabolic pathways required for independent survival in the marine environment. But bacteria that specialize in symbiotic relationships can afford to lose some of this genetic complexity, relying on their hosts to provide nutrients and protection that they would otherwise need to secure independently. This evolutionary streamlining often results in bacterial symbionts that are more efficient light producers, but less capable of surviving outside their partnership relationships. The optical properties of symbiotic luminescent systems demonstrate sophisticated integration between bacterial chemistry and host anatomy that produces lighting effects impossible for either partner to achieve independently. Bacterial colonies produce the raw illumination through their familiar luciferin-luciferase reactions, but host tissues shape and direct this light through biological optical systems that can create focused beams, diffuse illumination, or complex patterns of light and shadow. Some hosts develop multiple light organs with different optical properties, creating lighting systems with the versatility of complex engineered devices. The temporal control of symbiotic luminescence involves regulatory mechanisms that must coordinate bacterial metabolism with host behavior across timescales ranging from milliseconds to seasonal cycles. For rapid responses, such as the flash displays used to startle predators or signal to mates, hosts can modulate bacterial light output through neural signals that alter the chemical environment within light organs. For longer-term adjustments, hormonal systems can regulate bacterial populations and their metabolic activity in ways that match luminescent capacity to seasonal changes in feeding, reproduction, and migration patterns. Yet the maintenance of these partnerships requires constant molecular diplomacy between species with fundamentally different evolutionary interests. Bacteria benefit from maximizing their own survival and reproduction, which might involve strategies that conflict with their hosts' needs for controlled, efficient luminescence. Hosts benefit from maintaining bacterial partners that provide reliable illumination without excessive metabolic demands or harmful side effects. The balance between these sometimes competing interests must be maintained through chemical communication systems that have evolved to serve both partners' long-term survival. The geographic distribution of symbiotic luminescence partnerships reflects both the evolutionary history of these relationships and the ecological conditions that favor their development. In environments where bioluminescence provides significant survival advantages, particularly in the deep sea, where darkness is absolute and biological light represents the only available form of illumination, symbiotic arrangements have evolved repeatedly and diversified into numerous specialized forms. In environments where light is abundant or where bioluminescence provides fewer benefits. Such partnerships are less common and less elaborate. The metabolic integration between hosts and bacterial symbionts creates biological systems that blur traditional boundaries between individual organisms and ecological communities. A luminescent squid housing millions of bacterial partners represents neither a single organism nor a simple collection of independent creatures, but something intermediate, an integrated biological system that functions as a coherent unit while maintaining the genetic independence of its constituent species. Only took a few 100 million years for evolution to invent biological corporations. The biochemical waste management in these systems requires sophisticated mechanisms for disposing of the metabolic byproducts generated by bacterial light production without poisoning either the symbionts or their hosts. Bacterial luminescence produces not just photons, but also various chemical compounds that must be processed, neutralized, or eliminated to maintain the optimal conditions required for continued light production. Hosts must evolve physiological mechanisms for managing these waste products while maintaining the delicate chemical balance that keeps their bacterial partners healthy and productive. In the patient depths of ancient seas, the first symbiotic partnerships between light-producing bacteria and their multicellular hosts continue to refine their molecular negotiations, creating biological lighting systems that combine the efficiency of bacterial chemistry with the sophistication of multicellular anatomy. These relationships illuminate not just the dark waters they inhabit, but the collaborative possibilities that emerge when different forms of life discover mutual advantage in sharing their specialized capabilities. The candle burns without fire, tended now by partnerships that span the boundaries between species, creating light through cooperation that neither partner could achieve alone. The Midnight Zone's architecture unfolds in the permanent darkness of Earth's deep oceans, where creatures craft elaborate light displays with the patient artistry of beings who have never known any illumination except what they create themselves. Here, thousands of meters below the reach of sunlight, in waters so deep that the pressure would crush most surface organisms into molecular fragments, life constructs its most sophisticated experiments with bioluminescence, living galleries of light that transform the ocean's most forbidding depths into realms of alien beauty. The organisms that inhabit these depths face challenges that require innovations in luminescent technology far beyond what their shallow water ancestors developed. In the Midnight Zone, darkness is not merely the absence of light, but an absolute condition that has persisted unchanged for billions of years. No organism here has ever experienced sunlight directly. No evolutionary pressure has ever favored the development of vision systems adapted to solar illumination. Instead, life in the Midnight Zone has evolved entirely around the production, detection, and interpretation of biological light. The anatomical structures these deep-sea organisms develop for bioluminescence represent some of the most sophisticated biological optical systems on Earth. Many deep-sea fish construct elaborate light organs called photophores that function as precisely engineered optical devices, complete with lenses, reflectors, and shutters that can focus, direct, and modulate their biological illumination with remarkable precision. These organs often contain bacterial symbionts whose light production is regulated by complex neural and hormonal systems that allow their hosts to control luminescence with the fine timing required for hunting, communication, and predator avoidance. The spatial arrangement of photophores across the bodies of deep-sea organisms follows principles that seem almost architectural in their sophistication. Some species develop photophores arranged in precise geometric patterns that create specific light signatures, biological equivalent of visual identification codes that allow individuals to recognize members of their own species across the vast darkness. Others position their light organs to create counter-illumination systems that match the faint downwelling light that penetrates from far above, effectively rendering them invisible to predators or prey looking up from below. The colors these deep-sea organisms produce reflect both the optical properties of seawater and the evolutionary history of visual systems adapted to biological illumination. Most deep-sea bioluminescence occurs in the blue portion of the spectrum, wavelengths that travel furthest through seawater with minimal absorption or scattering. But variations exist that hint at the complex visual ecology of these depths. Some organisms produce red light that remains invisible to most deep-sea predators, whose visual systems have evolved to detect only blue and green wavelengths. This red bioluminescence functions like a private lighting system, allowing its users to illuminate their surroundings without alerting potential threats to their presence. The biochemistry underlying deep-sea bioluminescence often involves the same fundamental luciferin-luciferase reactions that enable luminescence in all organisms. But these basic chemical pathways have been elaborated and refined in ways that optimize them for the specific challenges of life in permanent darkness. Some deep-sea organisms have evolved multiple types of luminescent chemistry within the same individual, using different chemical pathways to produce different colors or intensities of light for different purposes. Others have developed mechanisms for recycling their luminescent compounds with exceptional efficiency, minimizing the metabolic costs of maintaining complex lighting systems in environments where food is often scarce. The temporal patterns of deep-sea bioluminescence reveal communication systems of remarkable sophistication, with different species employing distinct patterns of flashes, pulses, and steady glows to convey specific types of information. Some deep-sea organisms use rapid sequences of flashes as mating signals, with males and females exchanging precisely timed light displays that allow them to locate and identify each other across hundreds of meters of dark water. Others employ slower, more sustained luminescence to mark territory or maintain social cohesion within dispersed populations. The energy management strategies these organisms employ reflect the harsh realities of life in environments where food availability can be extremely limited and unpredictable. Many deep-sea creatures have evolved mechanisms for storing luminescent substrates during periods of abundant food, then rationing their light production during lean times to ensure they can maintain essential biological functions like reproduction and predator avoidance. Some species can completely shut down their luminescent systems during extended periods of starvation, then rapidly reactivate them when conditions improve. The evolutionary history of deep-sea bioluminescence involves multiple independent colonizations of the deep ocean by organisms that originally evolved in shallower waters. As these ancestral forms adapted to life in permanent darkness, their luminescent systems underwent modifications that optimized them for the specific conditions of the deep sea. The result is a remarkable example of convergent evolution, with unrelated lineages independently developing similar solutions to the challenges of communication, hunting, and survival in the midnight zone. The ecological interactions mediated by bioluminescence in the deep sea create complex webs of predation, competition, and cooperation that depend entirely on biological light production. Predators use luminescent lures to attract prey, creating the biological equivalent of anglers' lights that exploit their victims' attraction to illumination. Prey species counter with various forms of defensive bioluminescence, including burglar alarm displays that attract the predators of their predators, and sacrificial luminescence that allows them to shed glowing body parts as decoys while escaping into the darkness. The physiological integration required to support these complex luminescent systems involves sophisticated regulatory networks that coordinate light production with other essential biological functions. Neural systems must process sensory information about the surrounding environment and generate appropriate luminescent responses. Circulatory systems must deliver nutrients to light-producing organs while removing metabolic waste products. Hormonal systems must regulate bacterial symbionts and coordinate their light production with host behavior patterns. Yet mysteries persist in the bioluminescence of the midnight zone that continue to challenge researchers despite decades of careful study. How do deep-sea organisms coordinate their light displays across the vast distances that separate individuals in the sparse populations typical of the deep ocean? What determines the specific patterns of photophore arrangement that characterize different species? And how do these patterns relate to the ecological niches they occupy? How do the extreme pressures and temperatures of the deep sea affect the biochemistry of light production? And what adaptations allow luminescent reactions to proceed efficiently under these harsh conditions? The answers remain partially hidden in the biology of organisms that are difficult to study in their natural environment and often impossible to maintain in laboratory conditions that replicate the extreme conditions of their deep-sea habitats. Each species that has successfully colonized the midnight zone carries within its genome and physiology the solutions to challenges that seem almost impossibly difficult, yet have been solved through the patient work of natural selection operating across geological time. The optical landscape of the midnight zone represents one of Earth's most extraordinary biological achievements, an entire ecosystem based on the production and interpretation of biological light, where every significant ecological interaction depends on bioluminescence and where organisms have evolved sensory and behavioral systems entirely adapted to a world illuminated only by living creatures. The architecture of this darkness consists not of structures built from stone or metal, but of patterns of light and shadow created by organisms whose luminescence transforms the ocean's most forbidding depths into galleries of alien beauty that exist nowhere else in the known universe. In the crushing depths of the midnight zone, life continues its patient experiments with the chemistry of cold fire, crafting light displays that serve functions we are only beginning to understand while maintaining the fundamental biochemical processes that have enabled bioluminescence since the first bacterial lanterns began to glow in Earth's ancient seas. Only took 1 billion years for evolution to construct entire ecosystems based on biological light, and in constructing them, life demonstrates once again that the most inhospitable environments often inspire the most remarkable innovations in the endless creativity of natural selection. Fireflies carry their underwater torches onto land in one of evolution's most audacious experiments, taking the ancient chemistry of marine bioluminescence and adapting it to function in the radically different environment of terrestrial ecosystems. These aerial alchemists represent something unprecedented in the history of biological light production, organisms that glow not in the supporting medium of water, where bioluminescence first evolved, but in the thin atmosphere of a world where light travels unimpeded and darkness offers no permanent refuge from the daily cycles of solar illumination. The transition from aquatic to terrestrial bioluminescence requires solutions to challenges that never confronted their marine predecessors. In water, luminescent organisms enjoy the support of a dense medium that helps distribute their light while protecting their delicate biochemical machinery from temperature extremes and desiccation. On land, fireflies must maintain their light-producing chemistry in bodies exposed to dramatic fluctuations in temperature and humidity, while ensuring their luminescence remains visible across the optical complexity of terrestrial environments filled with obstacles, reflective surfaces, and competing sources of illumination. The anatomical modifications that enable terrestrial bioluminescence involve remarkable innovations in both the chemistry and the packaging of light-producing systems. Firefly light organs, called lanterns, represent sophisticated biological optical devices that concentrate luminescent chemistry in specialized cells called photocytes, then shape and direct the resulting illumination through complex arrangements of reflective and refractive tissues. These organs can produce light intensities that seem almost impossible given their size. A single firefly can generate flashes visible across hundreds of meters, turning a creature smaller than a paperclip into a biological lighthouse capable of communicating across vast terrestrial distances. The biochemistry these terrestrial light-bearers employ builds upon the same fundamental luciferase reactions that enable all bioluminescence, but with chemical modifications that adapt the ancient marine chemistry to the challenges of aerial life. Firefly luciferin represents a distinct molecular architecture optimized for the specific conditions of terrestrial light organs. Unlike the coelenterazine used by many marine organisms, firefly luciferin can function efficiently in the higher oxygen concentrations typical of terrestrial environments while maintaining stability across the temperature ranges that characterize life on land. The temporal control of firefly luminescence demonstrates precision that far exceeds what most marine organisms achieve. While many aquatic creatures produce light in response to immediate stimuli, predator attacks, food opportunities, physical disturbance, fireflies have evolved the ability to generate precisely timed light displays that serve as complex communication systems. Males of many species produce species-specific flash patterns that encode information about their identity, location, and reproductive fitness. Females respond with their own flash patterns that allow successful males to locate and approach them across the visual complexity of terrestrial habitats. These communication systems represent some of the most sophisticated uses of bioluminescence in the natural world, involving neural circuits that must coordinate light production with flight behavior, sensory processing, and the complex timing required for successful reproduction. A male firefly must not only produce the correct pattern of flashes for his species, but time these displays to coincide with optimal environmental conditions, navigate through three-dimensional space while maintaining visual contact with potential mates, and adjust his behavior in response to the competitive displays of other males operating in the same habitat. The energy economics of terrestrial bioluminescence involve considerations that differ significantly from those governing marine light production. On land, fireflies face the challenge of maintaining luminescent capability while also supporting the metabolically expensive processes required for flight. Their flight muscles demand constant supplies of ATP for the rapid contractions that enable aerial locomotion, competing directly with the energy requirements of their light-producing organs. The fact that fireflies can simultaneously fly and produce bright flashes of light represents a remarkable achievement in metabolic efficiency and resource allocation. The optical environment of terrestrial ecosystems creates both opportunities and challenges for bioluminescent communication that do not exist in marine systems. On land, light travels without the absorption and scattering effects of water, allowing firefly signals to propagate across greater distances with less energy investment. But terrestrial environments also contain numerous obstacles: trees, rocks, vegetation, artificial structures that can block light paths and create complex patterns of shadow and illumination. Fireflies must navigate these optical challenges while maintaining effective communication with potential mates who may be located in visually obstructed locations. The evolutionary history of terrestrial bioluminescence involves the remarkable transition of organisms adapted to aquatic life to the radically different conditions of terrestrial ecosystems. The ancestral forms that gave rise to fireflies almost certainly evolved their luminescent capabilities in aquatic environments, then gradually adapted to semi-aquatic and eventually fully terrestrial lifestyles. This transition required not just modifications to their light-producing chemistry, but comprehensive changes to their physiology, anatomy, and behavior that enabled them to survive and reproduce in environments where water is scarce and conditions can change rapidly. The diversity of firefly species demonstrates the remarkable evolutionary creativity with which these organisms have explored the possibilities of terrestrial bioluminescence. Different species employ different flash patterns, colors, and timing strategies that allow them to maintain reproductive isolation while occupying similar habitats. Some produce brief, intense flashes that maximize visibility while minimizing energy expenditure. Others create longer, more sustained displays that may convey more complex information about individual quality or reproductive status. Still others have evolved luminescence that serves functions beyond mate attraction, including predator deterrence and territorial defense. The ecological interactions mediated by firefly bioluminescence create new categories of biological relationship that have no direct analogues in marine ecosystems. Firefly-watching birds have evolved visual systems optimized for detecting luminescent displays, turning the predator-prey dynamics of terrestrial ecosystems into complex games of optical hide-and-seek. Some spiders construct webs positioned to intercept fireflies attracted to artificial lights, while other predators have learned to exploit firefly mating displays to locate and capture both males and females during their most vulnerable moments. The seasonal and circadian rhythms that characterize firefly bioluminescence reflect the temporal complexity of terrestrial environments, where day and night cycles create regular periods of illumination and darkness unknown in the permanent night of the deep sea. Most fireflies restrict their luminescent displays to specific times of day, typically during evening twilight periods when background light levels are low enough to make their flashes visible, but sufficient ambient illumination remains to support visual navigation. These timing strategies require internal biological clocks synchronized with environmental cycles that change predictably throughout the year. Yet mysteries persist in firefly bioluminescence that continue to intrigue researchers despite extensive study of these charismatic insects. How do fireflies coordinate their displays across populations that may span large geographic areas? And what determines the specific flash patterns that characterize different species? What role does individual variation in luminescent capability play in mate selection and reproductive success? How do environmental factors like temperature, humidity, and artificial lighting affect the chemistry and behavior of firefly bioluminescence? The cultural impact of fireflies on human societies represents something unique in the history of bioluminescence, the first luminescent organisms to achieve widespread recognition and appreciation among terrestrial species capable of aesthetic appreciation. Unlike the hidden bioluminescence of deep-sea creatures or the microscopic light of bacteria and dinoflagellates, firefly displays are readily visible to human observers and have inspired artistic, literary, and scientific interest across cultures worldwide. Only took 4 billion years for bioluminescence to develop an audience capable of conscious appreciation. In the gathering darkness of summer evenings, fireflies continue their aerial displays with the same chemical precision that has characterized bioluminescence since its earliest appearances in ancient seas. Their flashes illuminate not just their immediate surroundings, but the remarkable journey of biological light production from its aquatic origins to its terrestrial expressions. The candle that burns without fire has learned to fly, carrying the ancient chemistry of cold luminescence into environments that seem impossibly different from the dark waters where light-producing life first learned to glow, yet maintaining the fundamental biochemical processes that unite all luminescent organisms across the vast diversity of Earth's ecosystems. Evolution calculates the energy cost of photons with the mathematical precision of an accountant working in currencies of ATP and survival, balancing the metabolic expenses of light production against the reproductive benefits of visibility in an economy where every molecular transaction affects an organism's chances of passing its genes to future generations. The arithmetic of bioluminescence involves considerations that extend far beyond the simple chemistry of luciferin oxidation, encompassing the full spectrum of costs and benefits that determine whether the ability to produce light represents a profitable investment or an evolutionary luxury that organisms cannot afford. The direct metabolic costs of bioluminescence begin with the energy required to synthesize luciferin substrates, compounds whose molecular architecture demands significant investments of cellular resources. A typical luciferin molecule contains multiple carbon-carbon bonds, aromatic ring structures, and functional groups that must be assembled through multi-step biochemical pathways that draw on various pools of cellular metabolites. Each synthesis step requires enzyme catalysis, cofactor consumption, and the expenditure of ATP or other high-energy compounds that could otherwise be used for growth, reproduction, or basic cellular maintenance. The production of luciferase enzymes involves additional costs that compound the metabolic burden of luminescence. These proteins must be transcribed from DNA templates, translated by ribosomal machinery, and folded into precise three-dimensional configurations that enable their catalytic function. Many luciferases require post-translational modifications, the addition of metal cofactors, the formation of disulfide bonds, the incorporation of prosthetic groups that demand further cellular investment. Once synthesized, these enzymes must be maintained in functional condition, protected from oxidative damage, and eventually replaced when they become inactive due to normal molecular wear and degradation. The infrastructure required to support bioluminescence imposes additional costs that vary dramatically depending on the sophistication of an organism's light-producing systems. Simple bacteria that distribute luciferase throughout their cytoplasm face relatively modest infrastructure costs, but organisms that develop specialized light organs must invest in complex anatomical structures that house and support their luminescent chemistry. These organs often require specialized blood supplies to deliver nutrients and remove waste products, neural connections to control light production, and optical systems to focus and direct the resulting illumination. The opportunity costs of bioluminescence represent perhaps the most significant component of its true metabolic price. Every ATP molecule spent on luciferin synthesis is one less available for the synthesis of proteins needed for growth. Every amino acid incorporated into luciferase is one less available for the production of enzymes required for digestion, reproduction, or immune function. Every cellular resource devoted to maintaining light-producing capability is a resource that cannot be used for other biological functions that might contribute more directly to survival and reproductive success. Yet organisms across virtually every major branch of the tree of life have repeatedly evolved bioluminescent capabilities, suggesting that natural selection consistently finds the benefits sufficient to justify the costs. The advantages of light production must therefore outweigh its expenses by margins significant enough to overcome the conservative tendencies of evolutionary processes that typically favor metabolic efficiency over elaborate biochemical luxuries. The persistence of bioluminescence across geological time and biological diversity indicates that the ability to produce light provides advantages that more than compensate for its considerable costs. These benefits manifest in numerous forms that demonstrate the remarkable versatility of bioluminescence as an evolutionary strategy. For predators, luminescence can serve as a hunting tool that attracts prey or illuminates potential targets in dark environments where visual hunting would otherwise be impossible. For prey species, light production can function as a defensive mechanism that startles predators, attracts their enemies, or creates diversions that allow escape. For organisms seeking mates, bioluminescence provides a communication channel that can operate across distances far greater than those achievable through chemical or acoustic signals. The efficiency calculations that govern evolutionary investment in bioluminescence vary dramatically depending on environmental conditions and ecological context. In environments where light provides significant advantages, such as the deep sea, where darkness is absolute and biological illumination represents the only available light source, even relatively expensive luminescent systems prove cost-effective. In environments where light provides fewer benefits, such as shallow waters with abundant solar illumination, only the most efficient forms of bioluminescence justify their metabolic costs. The temporal economics of light production involve sophisticated cost-benefit calculations that organisms must perform continuously as environmental conditions change. Producing light during periods when it provides maximum advantage, such as during peak feeding times or reproductive seasons, represents efficient resource allocation. Maintaining luminescent capability during periods when it provides little benefit wastes resources that could be more profitably invested elsewhere. Many organisms have therefore evolved regulatory mechanisms that allow them to adjust their investment in bioluminescence based on changing circumstances. The evolutionary optimization of bioluminescent efficiency has produced remarkable innovations in the chemistry and regulation of light production that minimize costs while maximizing benefits. Some organisms have evolved mechanisms for recycling their luminescent substrates, capturing the molecular fragments produced by luciferin oxidation and reassembling them into fresh luciferin molecules. Others have developed systems for storing light-producing compounds during periods of abundant resources, then deploying them strategically when luminescence provides maximum advantage. The comparative economics of different approaches to bioluminescence reveal the diverse strategies organisms employ to balance costs against benefits. Some species invest heavily in continuous light production, maintaining steady illumination that provides constant advantages for hunting, navigation, or predator deterrence. Others concentrate their luminescent investment in brief, intense displays that maximize visibility while minimizing total energy expenditure. Still others adopt intermediate strategies that involve periodic or conditional luminescence triggered by specific environmental cues or behavioral contexts. The metabolic integration of bioluminescent systems with other cellular processes creates opportunities for efficiency improvements that reduce the net cost of light production. Many organisms couple their luminescent chemistry with other biochemical pathways in ways that allow shared use of enzymes, substrates, or metabolic intermediates. When luciferin synthesis pathways overlap with those used for other essential compounds, the marginal cost of light production decreases significantly. When luciferase enzymes can catalyze multiple types of reactions, the investment in their production serves multiple biological functions simultaneously. The scaling relationships between organism size and luminescent efficiency reveal additional complexities in the evolutionary economics of light production. Smaller organisms face higher metabolic costs per unit of light produced, but they also require less total illumination to achieve visibility across distances relevant to their survival and reproduction. Larger organisms can achieve greater efficiency in light production through economies of scale, but they must invest more total resources to illuminate their larger bodies adequately. These scaling relationships help explain why different types of organisms employ different strategies for bioluminescent investment. The social dimensions of luminescent economics emerge in species where individuals coordinate their light production to achieve collective benefits that exceed what any individual could accomplish alone. When bacteria engage in quorum sensing that synchronizes population-wide luminescence, the metabolic cost to each individual cell may be substantial, but the collective benefit of coordinated illumination justifies this investment. When fireflies synchronize their flashing to create large-scale displays, each individual bears the cost of precise timing coordination, but gains access to mating opportunities that would be unavailable through independent flashing. The coevolutionary dynamics between luminescent organisms and their predators, prey, and competitors create arms races that continuously reshape the cost-benefit calculations governing bioluminescent investment. As predators evolve improved visual systems for detecting luminescent prey, the prey must invest more resources in defensive bioluminescence or alternative survival strategies. As competing species develop more elaborate light displays for mate attraction, each species faces pressure to increase its own luminescent investment or risk reproductive disadvantage. The environmental factors that influence luminescent economics include not just the obvious considerations of ambient light levels and predation pressure, but subtle effects of temperature, water chemistry, and seasonal cycles that affect both the costs and benefits of light production. Higher temperatures increase the metabolic rates of luminescent reactions, while also increasing the energy demands of maintaining cellular function, creating complex interactions between thermal environment and luminescent efficiency. Changes in water chemistry can affect the stability of luminescent compounds and the efficiency of light-producing reactions in ways that alter the cost-benefit balance of bioluminescent investment. The evolutionary history of luminescent economics reveals long-term trends toward increased efficiency and more sophisticated cost-benefit optimization that reflect millions of years of natural selection pressure for metabolic efficiency. Early luminescent organisms likely operated with relatively inefficient systems that converted only small fractions of their energy input into useful light output. Through successive evolutionary refinements, modern bioluminescent systems have achieved efficiency levels that approach theoretical limits, converting up to 90% of their chemical energy directly into photons with minimal waste heat production. Yet fundamental constraints limit how efficient bioluminescent systems can become, creating evolutionary trade-offs that require organisms to balance competing demands for metabolic resources. The quantum mechanical properties that govern photon emission impose absolute limits on energy conversion efficiency that no amount of evolutionary optimization can overcome. The biochemical machinery required for luminescence must compete with other cellular systems for access to limited supplies of amino acids, cofactors, and metabolic intermediates. The regulatory mechanisms that control light production must themselves consume resources that add to the total cost of bioluminescent capability. The modern diversity of bioluminescent strategies reflects the different solutions various lineages have evolved to optimize their investment in light production under different environmental conditions and ecological pressures. Some have minimized costs by developing simple, efficient luminescent systems that provide basic functionality without elaborate infrastructure. Others have maximized benefits by investing heavily in sophisticated optical systems that can produce complex, highly visible displays. Still others have found intermediate solutions that balance moderate costs against moderate benefits in ways that prove optimal for their particular circumstances. The research challenges involved in measuring the true costs and benefits of bioluminescence continue to limit our understanding of its evolutionary economics despite decades of careful study. Quantifying the metabolic costs requires detailed knowledge of luminescent biochemistry and cellular energetics that is difficult to obtain for many species. Measuring the benefits requires understanding of ecological interactions and reproductive dynamics that often involve rare or difficult to observe behaviors. The long-term nature of evolutionary cost-benefit calculations means that short-term studies may miss crucial aspects of luminescent economics that only become apparent across generational time scales. In the patient calculations of natural selection, bioluminescence continues to prove its worth through the persistence and diversification of light-producing organisms across virtually every environment where life exists. The candle that burns without fire justifies its metabolic costs through advantages that extend far beyond simple illumination, creating communication channels, hunting tools, defensive mechanisms, and social coordination systems that transform the fundamental economics of survival and reproduction. Only took 4 billion years for evolution to perfect the accounting that makes biological light production profitable. And in perfecting these calculations, life demonstrates once again that the most successful innovations are those that provide benefits exceeding their costs by margins sufficient to sustain their elaboration across geological time. Camouflage through brilliance emerges as one of evolution's most counterintuitive strategies, where organisms achieve invisibility not by eliminating light, but by producing precisely the right amount of illumination to match the faint glow filtering down from far above. Counter-illumination represents a masterpiece of biological optics that transforms the physics of underwater light transmission into a sophisticated concealment technology, allowing creatures to disappear against the dim backdrop of downwelling radiance by becoming living sources of carefully calibrated luminescence. The physics underlying this remarkable strategy depends on the optical properties of seawater and the way sunlight behaves as it penetrates the ocean's depths. Solar radiation that reaches the sea surface undergoes dramatic transformation as it travels downward through increasingly dense layers of water. Blue wavelengths penetrate deepest, creating a faint azure glow that diminishes exponentially with depth but never disappears entirely. Even thousands of meters below the surface, red wavelengths are absorbed most rapidly, disappearing within the first few dozen meters. Green and yellow light falls somewhere between these extremes, creating a complex spectral environment that changes predictably with depth and time of day. For predators hunting from below, this downwelling light creates a natural detection system that silhouettes potential prey against the brighter waters above. Any creature swimming between a predator and the dim light filtering down from the surface appears as a dark shadow that stands out dramatically against the pale background. This optical disadvantage has shaped marine ecosystems for millions of years, creating selection pressures that favor any mechanism capable of eliminating or reducing the contrast between an organism's body and the surrounding illumination. Counter-illumination solves this problem through biological engineering of remarkable precision. Organisms that employ this strategy develop light-producing organs positioned on their ventral surfaces, the parts of their bodies that face downward toward potential predators. These photophores produce illumination that matches both the intensity and spectral characteristics of the downwelling light, effectively eliminating the silhouette that would otherwise make them visible to predators hunting from below. When viewed from underneath, a properly counter-illuminated organism becomes nearly invisible, its bioluminescent ventral surface blending seamlessly with the background illumination. The sophistication required for effective counter-illumination extends far beyond simple light production. The intensity of downwelling illumination changes continuously as organisms move vertically through the water column, requiring real-time adjustments to bioluminescent output that maintain optimal camouflage across different depths. The spectral composition of background light also varies with depth, time of day, and environmental conditions, demanding luminescent systems capable of producing light with precisely the right color characteristics for each situation. Many organisms achieve this precision through photosensitive control systems that continuously monitor ambient light levels and adjust their bioluminescent output accordingly. Specialized photoreceptors detect the intensity and spectral composition of downwelling illumination, then send signals to neural circuits that regulate the activity of ventral photophores. These biological control systems operate with feedback precision that rivals engineered optical devices, maintaining optimal counter-illumination across the wide range of conditions organisms encounter as they migrate vertically through the water column. The anatomical arrangements required for effective counter-illumination reveal remarkable innovations in biological optics that demonstrate sophisticated understanding of light physics encoded in genetic instructions refined across millions of years of evolution. Ventral photophores are often equipped with complex optical systems that include lenses, reflectors, and diffusers designed to create light distributions that precisely match the angular and spatial characteristics of background illumination. Some organisms develop multiple types of photophores with different optical properties, allowing them to create complex illumination patterns that eliminate shadows and provide uniform counter-illumination across their entire ventral surface. The biochemical systems that support counter-illumination must operate with exceptional reliability and efficiency, since any failure in light production immediately compromises camouflage effectiveness. Many organisms employ bacterial symbionts housed in specialized light organs that provide steady, controllable illumination without the metabolic costs of synthesizing luminescent compounds independently. These symbiotic relationships often involve sophisticated regulatory mechanisms that allow hosts to modulate bacterial light production in real-time response to changing environmental conditions. The evolutionary development of counter-illumination represents one of the most striking examples of convergent evolution in bioluminescent systems. Organisms from numerous unrelated lineages, fish, squid, crustaceans, and others, have independently evolved remarkably similar solutions to the problem of ventral concealment. This convergence suggests that counter-illumination provides such significant survival advantages that natural selection repeatedly favors its development despite the considerable challenges involved in achieving effective optical camouflage through biological light production. The ecological impact of counter-illumination extends far beyond the individuals that employ it, reshaping predator-prey dynamics throughout the marine ecosystems where it occurs. Predators that rely on silhouette detection must develop alternative hunting strategies when their prey becomes effectively invisible from below. This evolutionary pressure has led to the development of enhanced lateral line systems, improved chemical sensing capabilities, and hunting behaviors that exploit other types of sensory information. Some predators have evolved the ability to detect the slight thermal signatures of counter-illuminated prey, while others have developed visual systems sensitive to the subtle differences between bioluminescent camouflage and natural background illumination. The temporal dynamics of counter-illumination involve complex behavioral and physiological adjustments that must coordinate light production with other aspects of an organism's biology. Many species that employ counter-illumination are most active during specific periods when lighting conditions favor their camouflage strategy. Dawn and dusk represent particularly favorable times, when background illumination levels are low enough to make bioluminescent camouflage energy efficient but sufficient to provide meaningful concealment benefits. During periods of bright surface illumination, maintaining effective counter-illumination would require light production intensities that prove metabolically expensive, leading many organisms to seek deeper waters or remain inactive until conditions improve. The precision required for effective counter-illumination creates vulnerabilities that some predators have learned to exploit. Slight mismatches between bioluminescent output and background illumination can make counter-illuminated prey more visible rather than less visible, creating detection opportunities for predators with sufficiently sensitive visual systems. Environmental conditions that change rapidly, such as those associated with weather patterns or water movements, can compromise camouflage effectiveness if organisms cannot adjust their light production quickly enough to maintain optimal matching with background conditions. The energetic costs of maintaining counter-illumination systems involve not just the direct metabolic expenses of light production, but the additional costs of the sophisticated control and optical systems required for effective camouflage. The photosensitive monitoring systems that track background illumination must be maintained and calibrated. The neural circuits that process visual information and control photophore output require constant metabolic support. The optical elements that shape and direct bioluminescent output must be constructed from specialized biological materials that often involve significant synthetic costs. Yet the persistence and elaboration of counter-illumination across numerous marine lineages demonstrates that these costs are consistently justified by survival benefits that prove decisive in the evolutionary competition between predators and prey. The ability to achieve near invisibility through biological light production represents such a significant advantage that organisms continue to invest in increasingly sophisticated counter-illumination systems despite their considerable metabolic and developmental costs. The research challenges involved in studying counter-illumination reflect both the technical difficulties of measuring bioluminescence in natural marine environments and the complexity of the optical phenomena involved. Quantifying the effectiveness of biological camouflage requires simultaneous measurements of bioluminescent output, background illumination, and predator visual sensitivity under conditions that closely replicate the natural environment. The three-dimensional nature of marine light fields adds additional complexity, as effective camouflage must account for illumination arriving from multiple directions and varying in intensity and spectral composition across space and time. Modern technology has begun to reveal the extraordinary sophistication of counter-illumination systems through high-resolution imaging, spectroscopic analysis, and mathematical modeling of underwater light propagation. These studies confirm that many organisms achieve camouflage effectiveness that approaches theoretical limits, producing bioluminescent displays that match background illumination with precision that seems almost impossibly exact, given the biological constraints under which these systems must operate. In the patient depths where predators and prey engage in evolutionary arms races played out across millions of years, counter-illumination continues to demonstrate the remarkable innovations that emerge when natural selection encounters fundamental physical challenges. The strategy of hiding through illumination rather than darkness represents one of biology's most elegant solutions to the problem of concealment. Transforming the physics of light transmission into camouflage technology that allows organisms to achieve invisibility by becoming exactly bright enough to disappear. Only took hundreds of millions of years for evolution to discover that sometimes the best way to hide is to glow with precisely the right intensity. And in making this discovery, life once again demonstrates that the most successful adaptations often involve counterintuitive solutions that transform apparent disadvantages into sophisticated competitive advantages. The language of light develops as bioluminescence becomes communication, transforming the ancient chemistry of cold fire into sophisticated signaling systems that allow species to develop dialects written in photons rather than sound waves or chemical compounds. In the vast darkness of the deep sea and the optical complexity of terrestrial environments, organisms discover that light provides a communication channel capable of transmitting information across distances and through conditions that would defeat most other forms of biological messaging. The result is the evolution of luminescent languages whose grammar consists of flash patterns, color variations, and temporal sequences that encode meaning in streams of precisely controlled photon emissions. The fundamental advantages of photonic communication become apparent when compared to the alternatives available to organisms living in environments where other signaling methods face significant limitations. Chemical signals, while highly specific and capable of carrying complex information, diffuse slowly through water or air and can be disrupted by currents. Wind, or the chemical background noise generated by other organisms. Acoustic signals travel efficiently through many environments, but require specialized anatomical structures for production and reception, and can attract unwanted attention from predators or competitors. Visual signals based on reflected light depend on external illumination sources and fail completely in dark environments. Bioluminescent communication circumvents these limitations by providing organisms with their own portable light sources that function independently of environmental conditions. A luminescent signal can be produced instantly when needed, transmitted across considerable distances through dark water or air, and detected by any organism equipped with appropriate visual systems. The temporal precision possible with bioluminescent signaling allows for complex encoding schemes that can transmit detailed information about identity, location, reproductive status, and behavioral intentions through carefully orchestrated sequences of flashes, pulses, and sustained illumination. The diversity of luminescent languages that have evolved across different species reflects the remarkable flexibility of light as a communication medium. Some organisms employ simple binary codes based on the presence or absence of luminescence, basic on-off signaling that can convey fundamental information like species identity or reproductive readiness. Others develop more sophisticated systems that encode information in the duration of individual flashes. The intervals between flashes, or the intensity variations within continuous luminescent displays. Still others exploit the spectral dimensions of light, using different colors to convey different categories of information or to enable multiple simultaneous conversations through wavelength division multiplexing. The temporal structures of bioluminescent languages often mirror the communication requirements of the species that employ them. Fast-swimming predators that encounter potential prey or threats at high relative velocities require rapid signaling systems that can transmit essential information within brief windows of opportunity. Their luminescent languages typically consist of brief, intense flashes arranged in quickly recognizable patterns that maximize information transmission rates while minimizing the time spent in vulnerable signaling behaviors. Slower-moving organisms, or those that maintain stable territories, can afford more leisurely communication protocols that involve longer, more complex luminescent displays capable of conveying detailed information about individual quality, territory boundaries, or social relationships. The grammar underlying these luminescent languages reveals sophisticated information, encoding schemes that demonstrate remarkable computational sophistication for systems that evolved without any conscious design process. Many species employ hierarchical signaling structures where individual flashes combine into recognizable phrases, phrases combine into complete messages, and messages can be modified through variations in timing, intensity, or repetition patterns that convey additional layers of meaning. Some organisms develop contextual signaling systems where the same luminescent pattern carries different meanings depending on the environmental or social circumstances in which it is produced. The development of species-specific luminescent dialects creates reproductive isolation mechanisms that enable closely related organisms to maintain separate evolutionary trajectories even when they share similar habitats. Male fireflies of different species often employ flash patterns that are sufficiently distinct to prevent cross-species mating attempts, while remaining similar enough to suggest common evolutionary origins. These dialect differences can evolve rapidly when populations become geographically separated, leading to reproductive incompatibility that marks the early stages of speciation processes driven by luminescent communication systems. The reception and processing of luminescent signals requires visual systems that have evolved specifically to detect and interpret biological light sources. Many organisms that rely on bioluminescent communication develop enhanced visual sensitivity in the spectral regions where their own species produces light, while maintaining reduced sensitivity to wavelengths commonly produced by other organisms. This specialized visual tuning creates private communication channels that allow intraspecific signaling to proceed with minimal interference from the luminescent noise generated by other species operating in the same environment. The neural processing required for luminescent language comprehension involves sophisticated pattern recognition systems that can distinguish meaningful signals from background bioluminescence, evaluate signal quality and authenticity, and generate appropriate behavioral responses based on the information content of received messages. Some organisms develop specialized neural circuits dedicated to processing specific types of luminescent signals, while others employ more general visual processing systems that learn to recognize and respond to relevant luminescent patterns through experience and development. The evolutionary arms races between signaling organisms and the predators or parasites that exploit their communication systems create constant pressure for the refinement and optimization of luminescent languages. Fireflies must balance the need for conspicuous mating signals against the danger of attracting predatory spiders, birds, or other organisms that hunt luminescent prey. Some species have evolved signaling strategies that minimize predation risk through rapid, unpredictable flash patterns or the use of wavelengths that are less visible to common predators. Others coordinate their signaling with environmental conditions such as vegetation density or background lighting that provide natural camouflage for their luminescent displays. The social dimensions of luminescent communication extend beyond simple mate attraction to encompass complex coordination behaviors that enable group hunting, collective defense, and social organization systems based entirely on light-mediated information exchange. Some deep-sea organisms employ luminescent signals to coordinate feeding behaviors across dispersed populations, using light patterns to communicate information about prey availability, predator presence, or optimal feeding locations. Others use bioluminescent displays to establish and maintain territorial boundaries, create dominance hierarchies, or coordinate migration behaviors that require precise timing across large groups of individuals. The environmental factors that influence luminescent communication include both the optical properties of the medium through which signals must travel and the background conditions that affect signal visibility and interpretation. In marine environments, the absorption and scattering characteristics of seawater determine the maximum effective range of luminescent signals and influence the optimal wavelengths for different types of communication. In terrestrial environments, atmospheric conditions such as humidity, temperature, and air movement can affect both the production and detection of bioluminescent signals, leading to temporal and spatial variations in communication effectiveness. The conservation implications of luminescent communication systems create unique challenges for protecting species that depend on light-based signaling for essential biological functions. Artificial lighting from human activities can disrupt bioluminescent communication by creating background illumination that reduces signal visibility or by directly interfering with the visual systems organisms use to detect and process luminescent messages. Light pollution represents a particularly serious threat to firefly populations, whose mating systems depend entirely on the ability to produce and detect species-specific flash patterns in relatively dark environments. The technological applications inspired by biological luminescent communication systems demonstrate the practical value of understanding these natural signaling mechanisms. Researchers studying firefly flash patterns have developed insights relevant to optical communication systems, while investigations of deep-sea bioluminescent signaling have contributed to advances in underwater optical communication technologies. The efficiency and reliability of biological light-based communication systems continue to inspire engineering applications in fields ranging from telecommunications to autonomous vehicle coordination. Yet, fundamental mysteries persist in the evolution and function of luminescent languages that continue to challenge researchers despite decades of intensive study. How do the neural mechanisms underlying luminescent signal production and recognition evolve to maintain coordinated communication systems across species that may be separated by large geographic distances? What role does individual variation in signaling capability play in mate selection and reproductive success? How do environmental changes affect the long-term stability of luminescent communication systems? And what mechanisms allow these systems to adapt to changing conditions while maintaining their essential communication functions? In the darkness, where organisms must find ways to reach across space and time to coordinate their essential biological activities, bioluminescent communication continues to demonstrate the remarkable information processing capabilities that emerge when natural selection encounters the challenge of enabling complex social interactions through light-based signaling. The languages written in photons represent some of evolution's most sophisticated achievements in biological engineering, transforming the simple chemistry of luciferin oxidation into communication systems that rival human technologies in their complexity and exceed them in their efficiency and elegance. Only took millions of years for organisms to develop fluency in the grammar of light, and in achieving this fluency, They create communication channels that connect individuals across the vast distances and environmental barriers that otherwise isolate living creatures in their solitary struggles for survival and reproduction. Fungi's forest foxfire illuminates the terrestrial night with a ghostly radiance that emerges from decay itself, transforming the death of trees into exhibitions of cold light that seem to violate the fundamental relationship between illumination and combustion. These luminescent fungi represent something unique in the history of bioluminescence, organisms that derive their energy not from photosynthesis or predation, but from the patient decomposition of dead organic matter, using the chemical energy locked in cellulose, lignin, and other structural compounds to fuel the same luciferin-luciferase reactions that enable light production in marine bacteria and fireflies. The ecological niche occupied by luminescent fungi creates conditions that seem almost specifically designed to favor bioluminescent strategies. These organisms inhabit the forest floor and the interiors of decaying logs, environments where darkness reigns even during daylight hours and where the dense canopy above filters out most solar illumination. In such perpetually dark habitats, any source of biological light represents a significant advantage for organisms that must locate suitable substrates, attract dispersal agents, or coordinate reproductive activities across the complex three-dimensional landscape of fallen branches, leaf litter, and decomposing wood. The biochemistry underlying fungal luminescence follows the same fundamental pathways that enable light production in all bioluminescent organisms, but with molecular adaptations that reflect the unique metabolic capabilities of organisms that derive their energy from decomposition rather than photosynthesis or predation. Fungal luciferin appears to be related to the compounds used by other luminescent species, but with structural modifications that optimize its function in the specific chemical environment of fungal cells. The luciferase enzymes that catalyze light production show similar patterns of evolutionary relationship and functional divergence, suggesting that fungal luminescence evolved from the same ancestral biochemical machinery that enables bioluminescence throughout the living world. The spatial patterns of fungal luminescence create some of the most spectacular displays of bioluminescence in terrestrial ecosystems, when conditions are optimal, typically during warm, humid periods when fungal metabolism operates at peak efficiency. Entire fallen logs can glow with steady green radiance that transforms forest landscapes into scenes of otherworldly beauty. The luminescence often follows the growth patterns of the fungal mycelia, creating networks of light that trace the pathways through which these organisms explore and exploit their wooden substrates. The intensity of fungal luminescence varies considerably depending on species, environmental conditions, and the stage of the fungal life cycle. Some species produce barely detectable glows that require dark adapted vision to perceive, while others generate illumination bright enough to read by on moonless nights. The most spectacular displays typically occur during periods of active spore production, when fungal organisms invest maximum energy in reproductive activities that may benefit from increased visibility to potential dispersal agents. The temporal patterns of fungal luminescence reflect both the metabolic rhythms of decomposer organisms and the environmental conditions that affect their growth and reproduction. Many luminescent fungi show circadian variations in their light output, with peak luminescence occurring during nighttime hours when temperature and humidity conditions favor active metabolism. Some species coordinate their luminescent displays with seasonal cycles of spore production, creating predictable annual light shows that coincide with optimal conditions for reproductive success. The ecological functions served by fungal luminescence remain partially mysterious despite considerable research interest in these phenomena. The most widely supported hypothesis suggests that fungal light serves to attract arthropods and other small animals that might serve as spore dispersal agents, carrying fungal reproductive propagules to new locations where they can establish successful colonies. The wavelengths typically produced by luminescent fungi fall within the sensitivity range of many nocturnal insects, supporting the plausibility of this explanation for the evolutionary maintenance of fungal bioluminescence. Alternative hypotheses for fungal luminescence include the possibility that light production serves defensive functions, deterring potential predators or pathogens that might otherwise damage fungal tissues. Some researchers have proposed that bioluminescence might function as a form of metabolic regulation, providing organisms with a mechanism for dissipating excess metabolic energy during periods of abundant substrate availability. Still others suggest that fungal light might serve communication functions, allowing dispersed parts of fungal colonies to coordinate their activities across the complex spatial scales typical of forest environments. The diversity of luminescent fungi demonstrates the remarkable evolutionary success of bioluminescence as a strategy for organisms that occupy the decomposer niche in terrestrial ecosystems. Species in numerous fungal lineages have independently evolved luminescent capabilities, suggesting that the ability to produce light provides consistent advantages for organisms that must operate in the perpetually dark microenvironments characteristic of decaying organic matter. This convergent evolution parallels the independent development of bioluminescence in multiple marine lineages, indicating that light production represents a generally advantageous biological strategy wherever darkness creates opportunities for organisms that can generate their own illumination. The biochemical challenges faced by luminescent fungi include maintaining optimal conditions for light-producing reactions within cellular environments that must also support the complex enzymatic processes required for decomposing recalcitrant organic compounds like cellulose and lignin. Fungal cells must balance their investment in luminescent chemistry against the metabolic demands of producing the hydrolytic enzymes needed to break down their substrates, creating resource allocation decisions that affect both their competitive ability as decomposers and their effectiveness as light producers. The environmental factors that influence fungal luminescence include temperature, humidity, oxygen availability, and the chemical composition of the substrates these organisms colonize. Optimal luminescence typically requires warm, moist conditions that also favor rapid fungal growth and active metabolism. Oxygen availability affects both the efficiency of substrate decomposition and the oxidative reactions that enable light production, creating complex interactions between respiratory metabolism and bioluminescent chemistry that influence the intensity and duration of fungal light displays. The cultural impact of luminescent fungi on human societies reflects the powerful aesthetic and psychological effects of encountering biological light in terrestrial environments where such phenomena are relatively rare. Folklores from cultures worldwide include references to mysterious lights in forests that correspond closely to descriptions of fungal luminescence, and these phenomena have inspired numerous artistic, literary, and scientific investigations. The term foxfire itself reflects the long human fascination with these ghostly lights that appear to emerge spontaneously from decaying wood. The conservation implications of fungal luminescence relate primarily to the habitat requirements of forest ecosystems that maintain the complex spatial and chemical conditions necessary for supporting diverse communities of decomposer organisms. Old-growth forests with abundant coarse woody debris provide optimal conditions for luminescent fungi, while simplified forest ecosystems dominated by single species or lacking sufficient dead wood may not support the diversity of luminescent species that characterize more complex habitats. The research challenges associated with studying fungal luminescence include both the technical difficulties of measuring bioluminescence in forest environments and the complex ecological interactions that determine the distribution and activity patterns of these organisms. Fungal luminescence often occurs sporadically and unpredictably, making systematic study difficult. The ephemeral nature of many fungal fruiting bodies adds additional complications, as luminescent displays may be visible for only brief periods during the annual life cycles of these organisms. modern molecular techniques have begun to reveal the evolutionary relationships among luminescent fungi and the genetic mechanisms underlying their light-producing capabilities. These studies suggest that fungal luminescence evolved independently multiple times within different taxonomic groups, but consistently through modifications of the same basic biochemical pathways that enable bioluminescence in other organisms. The conservation of luminescent chemistry across such diverse lineages demonstrates both the evolutionary antiquity of these biochemical systems and their fundamental utility for organisms that must function in dark environments. Yet fundamental questions persist about the ecology and evolution of fungal bioluminescence that continue to motivate research despite the practical challenges involved in studying these phenomena. What specific advantages does light production provide to decomposer organisms that justify its metabolic costs? How do environmental changes affect the distribution and activity of luminescent fungi? And what role do these organisms play in forest ecosystem functioning? How do the biochemical pathways underlying fungal luminescence interact with the complex enzymatic systems required for decomposing recalcitrant organic compounds? In the patient darkness of forest floors, where the chemistry of death supports the physics of light, luminescent fungi continue their quiet work of transforming the molecular architecture of dead wood into streams of cold photons that illuminate the hidden processes of decomposition and renewal. Their ghostly radiance emerges from the most fundamental biological processes, the recycling of organic matter that enables the continuation of life in terrestrial ecosystems, yet manifests itself through the same quantum mechanical phenomena that govern all forms of biological light production. Only took hundreds of millions of years for fungi to learn to extract light from the darkness of decay. And in learning this chemistry, they demonstrate once again that bioluminescence represents one of life's most versatile and enduring strategies for transforming chemical energy into the visible signals that connect organisms across the vast spatial and temporal scales of biological existence. The chemical archaeology of bioluminescence unfolds as scientists decode the molecular history written in luciferase enzymes across species, reading backwards through genetic sequences and protein structures to reconstruct the 4 billion-year journey of cold fire from its origins in ancient bacterial cells to its magnificent elaborations in modern organisms. This detective work operates at the intersection of biochemistry and evolutionary biology, using the tools of molecular analysis to decipher the ancestral relationships, evolutionary innovations, and biochemical constraints that have shaped the development of biological light production across the tree of life. The protein sequences of luciferase enzymes serve as molecular fossils that preserve evidence of evolutionary events that occurred millions or billions of years ago. Unlike conventional fossils, which capture only the hard tissues and gross anatomical features of ancient organisms, these molecular records retain information about the precise chemical mechanisms that enabled life's earliest experiments with light production. Each amino acid position in a modern luciferase sequence reflects the accumulated history of mutations, selections, and functional optimizations that have occurred since the enzyme first evolved the ability to catalyze the oxidation of luciferin compounds. The comparative analysis of luciferase sequences across different species reveals patterns of evolutionary relationship that illuminate both the common origins and the independent innovations that characterize bioluminescent systems. Some luciferases show clear evidence of descent from common ancestral enzymes, with sequence similarities that reflect their shared evolutionary history. Others appear to have evolved independently from different starting points, representing convergent solutions to the fundamental challenge of catalyzing light-producing chemical reactions with high efficiency and specificity. The structural biology of luciferase enzymes provides additional layers of information about their evolutionary history and functional mechanisms. X-ray crystallography, and other analytical techniques reveal the three-dimensional architectures that enable these proteins to position luciferin substrates and cofactors with the atomic precision required for efficient light production. The active sites of luciferases from different species show both conserved features that reflect the universal requirements of luminescent chemistry and variable elements that reflect species-specific adaptations to different environmental conditions or functional requirements. The phylogenetic analysis of bioluminescent organisms creates evolutionary trees that trace the relationships among species that have independently acquired the ability to produce light. These analyses reveal that luminescence has evolved dozens of times throughout the history of life, appearing independently in bacteria, dinoflagellates, conidarians, arthropods, fish, and numerous other lineages. The repeated independent evolution of bioluminescence suggests that the ability to produce light provides such significant advantages that natural selection has consistently favored its development whenever the appropriate biochemical and ecological conditions align. The molecular clock analyses applied to luciferase gene sequences provide estimates of when different bioluminescent lineages diverged from their non-luminescent ancestors. These timing estimates, calibrated against known fossil evidence and geological events, suggest that the evolution of bioluminescence spans virtually the entire history of life on Earth. The earliest bacterial luminescence likely evolved more than 3 billion years ago, while more recent innovations in deep-sea fish and terrestrial fungi occurred within the past few 100 million years. The biochemical diversity revealed by comparative studies of luminescent organisms demonstrates the remarkable creativity with which evolution has explored the chemical possibilities of light production. Different species employ variations of the basic luciferin-luciferase reaction that produce different colors, temporal patterns, and efficiency characteristics optimized for their specific ecological requirements. Some organisms use multiple types of luminescent chemistry within the same individual, creating light displays that combine different wavelengths or temporal patterns to achieve complex signaling or camouflage functions. The genomic organization of bioluminescent genes provides insights into how the molecular machinery of light production has been integrated into the broader genetic systems that control organism development and function. In some species, luminescent genes are clustered together in operons that allow coordinated regulation of all the components required for light production. In others, the relevant genes are scattered throughout the genome but linked through complex regulatory networks that ensure appropriate coordination of luciferin synthesis, luciferase production, and the accessory systems required for functional bioluminescence. The horizontal gene transfer events that have shaped the evolution of bacterial bioluminescence create particularly complex patterns in the molecular archaeology of light production. The genes encoding luminescent functions can be transferred between bacterial species through plasmids, transposons, and other mobile genetic elements, creating evolutionary relationships that do not follow the normal patterns of vertical inheritance from parent to offspring. These horizontal transfers have contributed to the rapid spread of bioluminescent capabilities among bacterial lineages and may help explain the remarkable diversity of bacterial luminescent systems. The regulatory evolution of bioluminescent systems reveals how organisms have developed increasingly sophisticated control mechanisms for their light production capabilities. Early luminescent organisms likely produced light constitutively whenever substrates and energy were available. But more advanced systems have evolved complex regulatory networks that can modulate light production in response to environmental conditions, developmental stages, and social interactions. These regulatory innovations have enabled the evolution of sophisticated communication systems, precise camouflage mechanisms, and energy-efficient lighting strategies that maximize the benefits of bioluminescence while minimizing its metabolic costs. The molecular mechanisms underlying the spectral diversity of bioluminescent systems demonstrate how small changes in protein structure can produce dramatic alterations in the functional properties of luminescent chemistry. Single amino acid substitutions in luciferase active sites can shift emission spectra by dozens of nanometers, transforming blue light into green or green into yellow through mechanisms that operate at the boundary between classical chemistry and quantum mechanics. These structure-function relationships provide insights into how evolutionary processes can fine-tune the optical properties of bioluminescent systems to match the specific requirements of different ecological niches. The metabolic integration of luminescent pathways with other cellular processes creates additional layers of complexity in the molecular archaeology of bioluminescence. The synthesis of luciferin substrates often involves modifications of existing metabolic pathways rather than the evolution of entirely novel biochemical routes, suggesting that luminescence typically emerges through the repurposing of existing cellular machinery rather than through the invention of completely new chemical systems. These metabolic connections help explain how complex bioluminescent capabilities can evolve relatively rapidly when the appropriate selective pressures and biochemical starting materials are present. The coevolutionary relationships between luminescent organisms and their bacterial symbionts create particularly intricate molecular histories that must be decoded through careful analysis of both host and symbiont genomes. The genes that enable hosts to maintain bacterial partners, regulate their light production, and integrate their luminescence with host behavior represent evolutionary innovations that arose specifically to support symbiotic bioluminescence. Similarly, the bacterial genes that enable successful colonization of host tissues and appropriate responses to host chemical signals reflect adaptations that evolved to support the symbiotic lifestyle. The molecular basis of bioluminescent circadian rhythms reveals how luminescent systems have been integrated with the fundamental timing mechanisms that regulate biological activities across daily cycles. The genes that control circadian light production often involve the same molecular clockwork that regulates other rhythmic biological processes, but with modifications that enable precise temporal control of luminescent chemistry. These circadian adaptations demonstrate how bioluminescent systems can be fine-tuned to operate optimally during specific environmental conditions or behavioral contexts. The evolutionary constraints that have shaped the development of bioluminescent systems become apparent through comparative analysis of the molecular mechanisms underlying light production across different species. Certain biochemical requirements, such as the need for oxygen in most luminescent reactions, or the requirement for specific cofactors like ATP or metal ions, have imposed consistent limitations on how bioluminescent systems can evolve. These constraints help explain why luminescent organisms have converged on similar solutions to the challenges of light production despite their independent evolutionary origins. The molecular evolution of substrate specificity in luciferase enzymes reveals how these proteins have been optimized to work with specific luciferin compounds while excluding potential substrates that might interfere with efficient light production. The active sites of luciferases show clear evidence of selection for precise molecular recognition that ensures optimal binding and positioning of their intended substrates while discriminating against chemically similar compounds that might compete for binding but fail to produce light efficiently. The ancient origins of the luciferin biosynthetic pathways provide insights into the metabolic context in which bioluminescence first evolved. Many of the enzymes involved in luciferin synthesis appear to be modified versions of proteins that originally served other metabolic functions, suggesting that the evolution of light-producing capability built upon existing biochemical infrastructure rather than requiring the invention of entirely novel enzymatic activities. This pattern of evolutionary repurposing helps explain how complex bioluminescent systems could have evolved within the time frames suggested by molecular clock analyses. The comparative genomics of bioluminescent and non-luminescent closely related species illuminates the specific genetic changes that enabled the evolution of light production capabilities. In some cases, the acquisition of bioluminescence appears to have required relatively few genetic modifications, while in others it involved more extensive reorganization of metabolic pathways and regulatory systems. These comparative studies provide insights into the evolutionary accessibility of bioluminescent adaptations and the types of selective pressures that favor their development. The molecular signatures of positive selection in bioluminescent genes reveal which aspects of luminescent systems have been subject to the strongest evolutionary optimization. Regions of luciferase genes that show evidence of rapid evolutionary change often correspond to functional domains involved in substrate binding, catalytic efficiency, or regulatory interactions, suggesting that these aspects of luminescent chemistry have been under particularly intense selection pressure. Conversely, regions that show high sequence conservation across species likely represent functionally critical domains where changes would compromise essential biochemical activities. The epigenetic regulation of bioluminescent gene expression adds additional complexity to the molecular archaeology of light production, revealing how environmental conditions and developmental programs can modulate luminescent capabilities without changing the underlying DNA sequences. Methylation patterns, chromatin modifications, and other epigenetic mechanisms can affect when and where bioluminescent genes are expressed, providing organisms with flexible control systems that allow them to adjust their light production capabilities in response to changing conditions. The molecular mechanisms underlying the loss of bioluminescence in some evolutionary lineages provide insights into the selective pressures and biochemical constraints that determine when luminescent capabilities are maintained or abandoned. Some species that have colonized environments where light production provides fewer benefits have evolved reduced or eliminated bioluminescent systems, often through the accumulation of mutations that disrupt luminescent gene function. These evolutionary losses demonstrate that bioluminescence, despite its remarkable capabilities, is maintained only when its benefits consistently outweigh its metabolic costs. The modern molecular tools available for studying bioluminescence continue to reveal new aspects of its evolutionary history and biochemical mechanisms. CRISPR gene editing techniques allow researchers to manipulate luminescent genes and test hypotheses about their function and regulation. Advanced protein engineering methods enable the creation of novel luciferase variants with improved properties for research and biotechnological applications. Whole genome sequencing projects provide comprehensive views of how luminescent capabilities are integrated with other aspects of organism biology. Yet fundamental questions persist in the molecular archaeology of bioluminescence that continue to drive research despite remarkable advances in analytical techniques and theoretical understanding. How do the quantum mechanical aspects of photon emission relate to the classical biochemistry of enzyme catalysis? And what molecular mechanisms ensure optimal coupling between chemical energy and light production? What role do protein dynamics and conformational changes play in the catalytic cycles of luciferase enzymes? How do environmental factors affect the molecular mechanisms of bioluminescence? And what adaptations allow luminescent systems to function effectively under diverse conditions? The integration of molecular, physiological, and ecological approaches to studying bioluminescence creates increasingly comprehensive pictures of how light-producing systems function and evolve. Modern research programs combine laboratory studies of purified enzymes with field observations of luminescent behavior, creating multidisciplinary investigations that illuminate both the molecular mechanisms and the ecological functions of biological light production. These integrated approaches reveal connections between molecular evolution and ecological adaptation that were invisible to earlier generations of researchers working within more narrowly defined disciplinary boundaries. In the patient work of decoding the molecular messages preserved in luminescent organisms across the tree of life, scientists continue to uncover the remarkable history of biological light production and its central role in the evolution of life on Earth. The chemical archaeology of bioluminescence reveals not just the mechanisms by which organisms produce light, but the evolutionary processes that have shaped all of biology through 4 billion years of innovation, adaptation, and refinement. The molecular fossil record written in luciferase sequences tells a story that spans from the earliest bacterial experiments with cold fire to the sophisticated optical systems of modern deep-sea creatures, demonstrating once again that the most profound biological innovations often emerge through the patient accumulation of small molecular changes that eventually transform the fundamental capabilities of living systems. Only took 4 billion years for evolution to write the complete handbook of biological light production in the language of DNA and protein sequences. And in writing this molecular library, life has preserved for future investigation the detailed history of one of its most elegant and enduring innovations. Modern ocean's living aurora continues to pulse through today's seas with the same 4 billion-year-old chemistry that enabled the first bacterial lanterns, creating displays of biological light that transform contemporary marine environments into liquid galaxies where countless organisms contribute their individual photons to collective illuminations that rival anything in Earth's geological past. The persistence of bioluminescence across such vast spans of evolutionary time demonstrates not just the fundamental utility of biological light production, but the remarkable stability of the biochemical systems that convert chemical energy into cold fire with efficiency that continues to inspire human technologies while maintaining their essential functions unchanged across geological epochs. The distribution of bioluminescent organisms in modern oceans reflects both the ancient origins of luminescent chemistry and the ongoing evolutionary processes that continue to refine and elaborate light-producing systems. In the surface waters where sunlight penetrates, bioluminescence serves primarily defensive and communicative functions, with dinoflagellates creating their spectacular bloom displays and various planktonic organisms using light to confuse predators or attract prey. In the twilight zone, where solar illumination fades to insignificance, counter-illumination camouflage reaches its greatest sophistication as fish, squid, and crustaceans employ precisely controlled ventral lighting to blend with the dim background radiance filtering down from above. The deep sea represents the apotheosis of bioluminescent evolution, where organisms have had hundreds of millions of years to perfect their light-producing capabilities in environments of absolute darkness. Here, bioluminescence serves virtually every conceivable biological function from predation to reproduction to social coordination, creating ecosystems where biological light represents the primary source of illumination and where visual systems have evolved entirely around the detection and interpretation of luminescent signals. The remarkable diversity of deep-sea bioluminescent organisms, from bacterial colonies that coat entire seafloor communities with living light to giant siphonophores whose bodies stretch for dozens of meters while pulsing with coordinated luminescent displays, demonstrates the endless creativity with which evolution continues to explore the possibilities of biological light production. The biochemical sophistication of modern bioluminescent systems represents the culmination of 4 billion years of molecular evolution and optimization. Contemporary luciferase enzymes achieve energy conversion efficiencies that approach theoretical limits, transforming chemical energy into photons with minimal waste heat production through quantum mechanical processes that operate with remarkable precision and reliability. The regulatory systems that control modern bioluminescence involve complex networks of genetic, hormonal, and neural mechanisms that can modulate light production with temporal precision measured in milliseconds and spatial resolution that allows organisms to create complex patterns of illumination across their bodies. The ecological integration of bioluminescence in modern marine ecosystems creates food webs and community structures that depend fundamentally on biological light production for their organization and function. Predator-prey relationships often revolve around the production, detection, and interpretation of luminescent signals, creating evolutionary arms races where improvements in light-producing capability drive corresponding adaptations in visual systems and hunting strategies. Symbiotic relationships between luminescent bacteria and their hosts have evolved into sophisticated partnerships that provide both species with advantages neither could achieve independently, demonstrating the collaborative potential that emerges when different organisms combine their specialized capabilities. The temporal patterns of modern marine bioluminescence reveal the influence of ancient rhythms and contemporary environmental conditions on light production across scales ranging from individual cellular cycles to global oceanic phenomena. Many organisms maintain circadian rhythms in their luminescent activity that reflect the daily cycles of predation, feeding, and reproduction that structure marine ecosystems. Lunar cycles influence the reproductive timing of numerous luminescent species, creating monthly patterns of intensified light production that can transform entire regions of ocean into temporary bioluminescent spectacles. Seasonal variations in temperature, nutrient availability, and biological productivity create annual cycles of luminescent activity that contribute to the broader ecological rhythms that characterize marine environments. The geographic distribution of bioluminescent phenomena in modern oceans reflects both historical biogeographic processes and contemporary environmental gradients that affect the survival and reproduction of light-producing organisms. Tropical waters support the greatest diversity of bioluminescent species, while polar regions exhibit specialized luminescent communities adapted to the extreme seasonal variations in light and temperature that characterize high-latitude marine environments. The vertical distribution of bioluminescence shows clear patterns related to light availability, pressure, temperature, and food supply, with different luminescent strategies predominating at different depths within the water column. The conservation status of bioluminescent organisms in modern oceans faces challenges from human activities that affect both the organisms themselves and the environmental conditions that support their luminescent capabilities. Ocean acidification alters the chemistry of seawater in ways that may affect the efficiency of luminescent reactions and the survival of organisms that depend on precise biochemical balances for their light production. Climate change creates shifts in temperature, oxygen availability, and nutrient distribution that can disrupt the ecological relationships that support bioluminescent communities. Pollution introduces chemical compounds that can interfere with luminescent chemistry or damage the organisms that produce biological light. The research techniques available for studying modern bioluminescence combine traditional fieldwork with advanced technologies that enable detailed observation and analysis of luminescent phenomena in their natural environments. Remotely operated vehicles equipped with sensitive cameras and spectrometers can document bioluminescent displays in the deep sea without disturbing the organisms that produce them. Laboratory culture systems allow controlled studies of luminescent biochemistry and behavior under conditions that replicate natural environments. Molecular techniques provide insights into the genetic and biochemical mechanisms underlying light production, while ecological modeling helps researchers understand the broader environmental factors that influence bioluminescent communities. The biotechnological applications derived from modern bioluminescent systems demonstrate the practical value of understanding biological light production for human technologies and medical applications. Luciferase enzymes isolated from various organisms serve as tools for molecular biology research, environmental monitoring, and medical diagnostics. Bioluminescent organisms provide model systems for studying fundamental biological processes like gene expression, cellular communication, and metabolic regulation. The principles underlying biological light production inspire innovations in optical engineering, energy conversion, and sustainable lighting technologies. The educational and cultural significance of modern marine bioluminescence continues the long human fascination with biological light that spans cultures and historical periods worldwide. Bioluminescent displays serve as powerful examples of the beauty and complexity of natural systems, inspiring scientific interest and environmental conservation efforts. The accessibility of some bioluminescent phenomena, such as the glowing wakes created by disturbed dinoflagellates or the seasonal appearances of bioluminescent blooms in coastal waters, provides opportunities for public engagement with marine biology and ecosystem science. The evolutionary future of bioluminescence in changing oceans remains uncertain, but will likely involve continued adaptation to environmental conditions that differ significantly from those that shaped the historical development of light-producing systems. Organisms with flexible luminescent capabilities may be better positioned to survive environmental changes than those with highly specialized light-producing systems optimized for specific conditions. The rapid pace of contemporary environmental change may favor organisms that can quickly modify their bioluminescent strategies through behavioral or physiological adjustments, rather than those that require genetic changes to adapt their light production capabilities. The interconnectedness of bioluminescent organisms with broader marine ecosystems means that changes in luminescent communities can have cascading effects on other aspects of ocean ecology. The loss of key bioluminescent species can disrupt predator-prey relationships, alter energy flow through marine food webs, and affect the ecological services provided by marine ecosystems. Conversely, the introduction of bioluminescent species to new environments, whether through natural range expansions or human-mediated transport, can create novel ecological interactions with unpredictable consequences for existing biological communities. The ongoing research into modern marine bioluminescence continues to reveal new species, biochemical mechanisms, and ecological functions that expand our understanding of the role biological light production plays in contemporary ocean ecosystems. Advanced deep-sea exploration techniques regularly discover new luminescent organisms in previously unexplored habitats, while molecular studies reveal previously unknown diversity in the biochemical mechanisms underlying light production. Long-term ecological studies document changes in bioluminescent communities over time, scales that illuminate both natural population dynamics and the effects of human activities on marine environments. Yet fundamental mysteries persist in the ecology and evolution of modern marine bioluminescence that ensure continued research interest despite decades of intensive study. How do global environmental changes affect the complex biochemical processes underlying light production? And what adaptive capacity do luminescent organisms possess for responding to rapidly changing conditions? What role does bioluminescence play in marine ecosystem functioning at scales ranging from individual organisms to ocean basins? How do the quantum mechanical processes that enable photon emission interact with the broader biological systems that support life in marine environments? In the vast expanses of modern oceans where biological light continues to pulse through dark waters with the same fundamental chemistry that enabled life's first experiments with luminescence 4 billion years ago, the living aurora demonstrates both the continuity and the creativity that characterize biological evolution across geological time. The persistence of bioluminescence through Earth's greatest environmental changes and biological innovations reveals something essential about the relationship between chemistry and life, showing how quantum mechanical processes can be harnessed by biological systems to create phenomena of remarkable beauty and functional sophistication. Only took 4 billion years for the candle that burns without fire to spread throughout Earth's oceans and adapt to every conceivable marine environment, and in maintaining its ancient chemistry while continuously exploring new possibilities. Bioluminescence demonstrates once again that the most successful biological innovations are those that combine fundamental chemical principles with endless evolutionary creativity to produce solutions that seem both inevitable and miraculous. The quiet burning continues as darkness envelops the world, and life maintains its cold fires with the patient persistence of chemistry that has never forgotten how to transform metabolic energy into streams of photons that cut through the surrounding blackness without generating a single degree of unnecessary heat. In every corner of Earth where organisms have discovered the advantages of biological illumination, the ancient machinery of bioluminescence operates with the same fundamental precision that enabled the first bacterial lanterns to glow in primordial seas, demonstrating through its endless persistence that some innovations transcend the temporary adaptations that characterize most evolutionary solutions and instead represent permanent additions to life's essential toolkit for survival and reproduction. Across the planet's diverse ecosystems, bioluminescent organisms continue their nocturnal activities with the quiet efficiency that characterizes biological processes refined across geological time. In tropical tide pools, bacterial communities coat the surfaces of rocks and algae with living films of light that pulse in response to wave action and the movements of nocturnal creatures seeking food or shelter. In temperate forests, luminescent fungi transform fallen logs into natural lighthouses that guide arthropods through the complex landscape of the forest floor while facilitating spore dispersal through mechanisms that remain partially mysterious despite decades of careful study. The deep ocean maintains its role as the planet's primary repository of bioluminescent diversity, where darkness has provided consistent selective pressure for light-producing capabilities since life first colonized the abyssal depths. Here, organisms continue to perfect lighting technologies that surpass human engineering achievements in their efficiency, reliability, and integration with biological systems. Schools of lanternfish coordinate their photophore displays to create moving constellations that migrate vertically through the water column with the predictability of celestial bodies, while solitary anglerfish maintain their bacterial lures with the dedication of lighthouse keepers tending beacons that guide prey to their destruction. The persistence of symbiotic relationships between light-producing bacteria and their multicellular hosts demonstrates the stability of cooperative strategies that have evolved to share the metabolic burdens and functional benefits of bioluminescence. These partnerships, refined over millions of years of coevolution, represent some of biology's most sophisticated examples of interspecies cooperation, with hosts providing carefully controlled environments for their bacterial partners while receiving in return precisely regulated illumination that can be modulated for hunting, communication, and predator avoidance with the precision of engineered optical systems. The seasonal rhythms that govern bioluminescent activities in many ecosystems reflect the integration of light-producing capabilities with the broader environmental cycles that structure biological communities across temporal scales, ranging from daily to annual. Spring blooms of dinoflagellates transform coastal waters into liquid fire as warming temperatures and increasing nutrient availability create optimal conditions for rapid population growth and intensified luminescent display. Summer evenings pulse with the coordinated flashes of fireflies, whose mating displays have become synchronized with temperature cycles, lunar phases, and the complex acoustic landscapes created by other nocturnal organisms competing for limited communication channels. The evolutionary arms races between luminescent organisms and the predators, prey, and competitors that respond to their light production continue to drive innovations in both light production and light detection that demonstrate the ongoing creativity of natural selection when faced with the challenges of survival in environments where biological illumination plays critical ecological roles. Predators develop enhanced visual sensitivity and behavioral strategies optimized for detecting and exploiting luminescent prey, while their targets evolve counter-strategies that include deceptive lighting, temporal coordination of illumination with predator activity patterns, and the development of luminescent signals that attract the predators of their predators. The biochemical stability of luminescent systems across such vast spans of evolutionary time reflects both the fundamental utility of the underlying chemical mechanisms and the constraints imposed by quantum mechanical laws that govern the relationship between molecular structure and photon emission. The luciferin-luciferase reactions that enable modern bioluminescence operate through the same basic chemical pathways that enabled the first luminescent bacteria, yet continue to be refined through subtle modifications in enzyme structure and substrate composition that optimize light production for specific ecological contexts and environmental conditions. The geographic distribution of bioluminescent organisms across Earth's diverse habitats demonstrates both the versatility of biological light production as an adaptive strategy and the environmental factors that favor its evolution and maintenance. Marine environments continue to support the greatest diversity of luminescent species, reflecting both the ancient origins of bioluminescence in aquatic systems and the consistent advantages that light production provides in environments where darkness predominates and visual communication offers significant benefits over chemical or acoustic alternatives. Terrestrial bioluminescence, while less diverse than its marine counterpart, continues to serve essential functions in forest ecosystems where light penetration is limited and organisms must rely on biological illumination for activities ranging from mate location to predator avoidance. The success of fireflies as terrestrial light producers demonstrates that the transition from aquatic to aerial bioluminescence, while challenging, opens new ecological opportunities that justify the considerable physiological and biochemical adaptations required for maintaining light-producing capabilities in environments where water is scarce and temperature fluctuations are extreme. The research frontiers that continue to expand our understanding of bioluminescent systems reveal both the remarkable sophistication of biological light production and the fundamental mysteries that persist despite centuries of scientific investigation. Modern techniques for studying quantum mechanical processes in biological systems illuminate the molecular mechanisms that enable such efficient conversion of chemical energy to light, while ecological research reveals the complex community-level interactions that both depend on and influence bioluminescent activities across diverse ecosystems. The conservation implications of maintaining bioluminescent diversity in rapidly changing environments create challenges that require understanding of both the immediate threats facing light-producing organisms and the broader ecosystem processes that support their long-term survival. Climate change, pollution, and habitat destruction affect bioluminescent communities in ways that can disrupt ecological relationships developed over millions of years. While the increasing prevalence of artificial lighting creates novel environmental conditions that interfere with the light-based communication systems many organisms depend on for reproduction and survival, the technological applications inspired by biological light production continue to expand as human understanding of luminescent biochemistry advances and engineering capabilities develop to match the sophistication of natural systems. Bioluminescent proteins serve increasingly important roles in medical diagnostics, environmental monitoring, and fundamental research. While the principles underlying biological light production inspire innovations in sustainable lighting, energy conversion, and optical communication systems that may eventually rival the efficiency and elegance of their biological inspirations, the aesthetic and cultural significance of bioluminescence continues to inspire human artistic and scientific endeavors that celebrate both the beauty and the functional sophistication of biological light production. The visual spectacle created by bioluminescent displays serves as a powerful reminder of the creativity and elegance that characterize natural systems while providing accessible examples of the complex biochemical and ecological processes that sustain life on Earth. Yet the true significance of bioluminescence extends beyond its immediate practical applications or aesthetic appeal to encompass its role as one of the evolution's most persistent and successful innovations. The ability to produce light has emerged independently dozens of times throughout the history of life, appearing in organisms as diverse as bacteria and beetles, fungi and fish, demonstrating that the advantages of biological illumination consistently outweigh its metabolic costs across a remarkable range of environmental conditions and ecological contexts. The quantum mechanical foundations of bioluminescence connect the molecular processes that enable light production with the fundamental physical laws that govern energy and matter throughout the universe, revealing how biological systems have learned to harness quantum phenomena for practical purposes millions of years before human scientists developed the theoretical frameworks needed to understand these same processes. The precision with which biological systems control photon emission demonstrates a mastery of quantum mechanics that emerges from evolutionary optimization rather than conscious design, yet achieves results that seem almost impossibly elegant and efficient. In the gathering darkness that envelops Earth each evening, bioluminescent organisms across the planet begin their nocturnal activities with the quiet confidence of chemistry that has proven its worth across 4 billion years of evolutionary testing. Their cold fires burn without smoke or flame, without noise or waste, transforming the chemical energy of life into streams of photons that serve functions ranging from the prosaic to the spectacular, while maintaining the fundamental biochemical principles that have enabled biological light production since life first learned to glow in ancient seas. The candle that burns without fire continues its patient illumination of the world's dark places, tended by organisms that know nothing of quantum mechanics or biochemistry or evolutionary theory, yet maintain through their living chemistry the most elegant demonstration of how biological systems can harness fundamental physical processes to create phenomena that seem to transcend the ordinary limitations of matter and energy. Only took 4 billion years for life to perfect the art of cold combustion, and in perfecting this ancient chemistry, organisms across Earth continue to demonstrate that the most profound innovations often involve not the creation of something entirely new, but the endless refinement of solutions that prove so fundamentally useful that they persist unchanged in their essential characteristics while adapting endlessly to the changing circumstances that define existence in a dynamic world. As the deep currents of geological time carry the present moment toward futures unimaginable, the eternal ember of bioluminescence settles into the quiet rhythms that have sustained it across 4 billion years of planetary change. The urgent chemistry of luciferin oxidation slows to match the patient heartbeat of cosmic processes, each photon emission becoming part of a larger pattern that connects the briefest quantum mechanical transitions with the vast temporal scales on which evolution operates its most fundamental transformations. In the deepest ocean trenches where pressure creates conditions more extreme than the surface of other planets, bacteria continue to glow with the same molecular precision that enabled the first luminescent cells in Earth's ancient seas. Their light represents not just a biological function, but a form of cosmic memory, the preservation of chemical knowledge that spans the gap between the earliest experiments in organized matter and the most sophisticated achievements of contemporary life. The quantum mechanical processes that enable their illumination operate according to physical laws that predate the formation of planets, yet find expression through biological systems so refined that they seem to represent the inevitable outcome of chemistry, exploring its own possibilities across unlimited time. The rhythm of photon production gradually synchronizes with larger patterns, the circulation of deep ocean currents that move water masses across entire hemispheres, the precession of Earth's axis that creates cycles of climate change spanning tens of thousands of years, the orbital variations that govern ice ages and warming periods across millions of years. Bioluminescence becomes part of the planet's longest conversations with itself, a dialogue conducted in the language of cold fire that connects the molecular scale of individual reactions with the planetary scale of global biogeochemical cycles. The ancient chemistry continues its work as consciousness fades and the boundaries between individual organisms and the larger systems they inhabit begin to dissolve. Bacterial colonies merge into continuous films of living light that coat the surfaces of deep-sea sediments. Fungal networks spread their luminescent mycelia through forest soils, in patterns that mirror the distribution of neural networks in sleeping brains. The light of fireflies becomes indistinguishable from the light of stars, both representing the conversion of simple energy differences into the photons that make existence visible to any observer capable of detecting their presence. In this deepest darkness, where all individual purposes fade into the background patterns that sustain life across geological time, bioluminescence reveals its most fundamental nature as a bridge between the living and the non-living world. The same quantum mechanical principles that govern stellar fusion and galactic formation find expression in the molecular architectures that enable biological light production, creating continuities that span from the cosmic to the cellular scale. The candle that burns without fire becomes indistinguishable from the fires that burn in the hearts of distant suns, all part of the universal tendency of energy to organize itself into patterns that persist, evolve, and create new possibilities for complexity and beauty. The eternal ember settles into its final rhythm, the slow pulse of planetary breathing, the gentle circulation of elements through living systems and back to the geological processes that created them. The patient exchange of energy between matter and light that sustains all the elaborate architectures of organized chemistry that we recognize as life. In this ultimate stillness, bioluminescence continues its quiet work of demonstrating that existence itself is a form of controlled combustion, a way of transforming the basic energy differences that define physical reality into the patterns of organization and beauty that make consciousness possible. Sleep well, knowing that even in the deepest darkness, the ancient chemistry of cold fire continues its patient work, tending the molecular flames that connect each living moment with the 4-billion-year history of biological innovation and the unlimited future of possibilities that await discovery by forms of life and consciousness that have not yet learned to dream of their own existence.